উনিদলে জয়েন করেছে। আমাদের ঢুকতেই দিচ্ছে না, তার আগে পৌঁছে যাচ্ছে। তাহলে কি করে করবো আমি বিনা এন্ট্রি? 7টা বাজে 2 মিনিট দিয়ে আবার আগে রেকর্ডিং শুরু করি, ঠিক আছে? নোটিশ হয়েছিল। আমি এখান থেকে বলি সব কাজ অসুবিধা কিছু হয়নি, আমি এখান থেকে বলি অসুবিধা কিছু হবে নি তো? হ্যাঁ, ডিস্টার্ব হবে না? হ্যাঁ, দেখি ভিডিওটা খুলি। পিছনে একটা বন্ধ হয়েছে, টিউবলাইটটা বন্ধ হয়েছে। পিছনে টিউবলাইট বন্ধ করতে হবে। পিছনে টিউবলাইটটা বন্ধ করতে হবে। বন্ধ কর। তুই তো মোবাইল থেকে ছবি দিতে পারবি না রে? ছবি ছবি ক্যামেরা। না, আমি একটু সরে বসি। ঘুরে বলছি কম হবে। লাইটটা একটু দে। ওইটা অফ করে দিই। অন্ধকার আছে, আটকে লাইট এই অ্যাঙ্গেলে দিলে কেমন হবে? দেখা যাচ্ছে না। ওইটাই ঠিক আছে, ওটা অফ করে দিই। চশমাটা আমার ধরে কি করে করবো? লঙ্কা করে দিলাম, ভোলা লাগে। ফোন ধরবি না।
Hello, Devbabu. Good evening.
Good evening.
Good evening. Good evening, Dept sir.
Good
Good
evening,
evening,
sir.
sir.
Prabhulla sir,
Prabhulla
good
sir, good
evening.
evening.
Prabhulla
Prabhulla
sir,
sir,
good
good
evening.
evening.
Good evening, Dr. Das.
Actually,
Actually,
today
today
is
is
raining.
raining.
আর এটা হচ্ছে না না, ছবি ছবি একটা নেবো, এটা এটা দুটাকে টেবিলে তো হয়। একটা ধরো ছবি আগে হয়। ঠিক আছে, পপুলার থাকা মানে আরাম সন্ধি হয়ে গেছে রিসার্চ করছি। না ছোট, হুম,
আমার 14 আছে হয়ে যাবে। আমার তো কত সময় লাগবে।
এটা চালু করছি রেকর্ডটা। এটা তো নাই, ভয়েসটা না খুললে তো রেকর্ড হবে না, ভয়েস, তাই না?
ভয়েস অফ করা আছে।
কোনটা?
আপনার ভয়েস অফ করা আছে
না, এটা খুলে দিলে তো রেকর্ড হবে নাকি?
হ্যাঁ।
শুরু করি, 7 জায়গা গেল নাকি? শুরু করছি, বুঝলে? শুরু করুন। 7টা 1 হয়ে গেল, করে দিই আর কি। দই পান ঢুকছে কিনা দেখি একটু। দই পান এখন ঢোকেনি, ফলে শুরু করে দিই নাকি, না কি করবো? শুরু করছি নাকি? শুরু করতে দই পান ঢুকবো, তখন তারপরে যা হবে। ও, দেরি করে লাভ নেই। So, Dept sir, are you here? Our conveners, webinars, seminars, public publication subcommittee. Dept with sir. Just waiting 2 minutes. Sir will join, and after that we will start. Dept sir, can we start?
Yes, sir, sure.
So, good evening to one and all in this virtual platform. Myself, Dept of Botany, Associate Professor of Botany and IQC Coordinator, Government General Degree College, Lalgarh. Previously, it was synonymously used Lalgarh Government College under West Bengal Government Higher Education Department. We are going to arrange day 2 program through e-mode. You know, on 20th already we organized a program, and in this program, earlier date 4 resource persons are there. Today we have 3 resource persons. Dr. Profullo Seti, Department of Microbiology, MA Abasaheb Garwani College, Pune, Maharashtra State. Its title is Exploring Role of Microbes in Cell Healing, Concrete Revolutionary Approach. Second, invited talk by research person from Institute of Forest Genetics and Trade Breeding Institute. She is scientist Dr. D. Thangamani from Coimbatore, Tamil Nadu. Her title, Characterizations of Genetic Resource Using Molecular Markers. Third, resource person from our state. He is my friend Dr. A. Chondo, UG and PG Department of Zoology, Raja N.L. Khan Women's College, Mindapur, West Bengal. His title is Aquatic Ecosystem, Plants, Animals and Microbial Association for Ecosystem Functioning. This actually program we just arranged in such a sequence, 3 invited talks, followed by vote of thanks by one professor and head department of life science from SAM College, Bhopal, Madhya Pradesh, Dr. Rajani Gautam. After that, we will move to the technical session for rest of the participants to be serially called for discussion. That is serial number 21 onwards. No presentation today like the previous day. So this session to be delivered by the scientists, by the researchers, by the scholars, UG and PG students and so on, and directed by Dr. Vinutar Bhatto, Department of Zoology and Genetics, Jyoti Nivas College, Bangalore, West Bengal. In the panel discussion forum, Some teachers are here. I think some are joined and some are willing to join shortly. Dr. Vidyanath Jha, our retired professor and principal, Ellen Mithila University, Darbhanga, Bihar. Sir is also our linkage group member. Delivered many lectures on different fields. Professor Dr. Ravindranath Bhattacharya, retired professor of Life Science, Presidency University, Kolkata. Sir from microbiology, Dr. Dr. Majita Begum, Dr. A. Majita Begum, Head Departments of Botany, Haji Karutha, Rauthar, Howdia College, Uthama Palam, Theni, Tamil Nadu. Dr. G. Maheshwari, Departments of Zoology, Abhinali Singham, University, Tamil Nadu. Dr. V. Gayatri, Departments of Botany, Abhinali Singham, University, Tamil Nadu. Dr. Ravi Kumar Misro, Departments of Biodiversity Conservation, Maharaja Bhanjdev University, Takotpur, Mayurbhanj, Odisha. Previously, it was North Odisha University in Odisha. Dr. Pompi Ghosh, Head Departments of Botany, Seva Bharati Mahavidyalaya, Kapgadi, Jhargram, West Bengal. Mr. Santosh Murmu, Assistant Professor of Botany, Raja Enel Khan Women's College, Paschim Medinipur, West Bengal. Dr. Vireshwar Bera, Department of Zoology, St. Joseph's College, Darjeeling, West Bengal. Dr. Sumitra Salam, Assistant Professor, Department of Botany, Nambol L. Sanoi College, Nambol, Imphal, Manipur. Dr. K. Mahajan, Head Departments of Zoology, GGDC, sorry, Dr. K. Maji, Head Departments of Zoology, GGDC Lalgarh, Jhargram. Dr. Sujit Kisku, Assistant Professor of Zoology, GGDC Gopivallabhpur, Piliyabada, Jhargram. Dr. Bhuisali P. Bamsod from Maharashtra, Pune, and so on. So already we mentioned some panel experts for day one. This is the day two list, and this list is going to be infinitive. Because already we received many CV to prepare the reviewers panel, because we will launch one journal shortly in the year 2025. So shortly I am coming to this session of conference day 2 in this program. Now Dr. Pompi Ghosh, Assistant Professor, I request Professor Ghosh to say about something about the conference. Dr. Ghosh, are you here?
Yes, sir.
Dr. Ghosh is also a mentor teacher in our college, because Mau College of Seva Bharati Mahavidyalaya, GGDC Lalgarh, already established, and departmental Mau already established. So she is one guest faculty of our college also. Dr. Ghosh, now the platform is yours.
Thank you, sir. Good evening to all. Myself, Dr. Pompi Ghosh, Assistant Professor and Head, Department of Botany, Shivavarati Mahavidyalaya, Kapgadi, Jhargram. On behalf of the General Member, National Seminar Organizing Committee, welcome all of you to this virtual room for secondary program. The theme of the conference, as you know, is interdisciplinary approach on plant, animal science, and microbes. Nowadays, we are very much associated with bioprospecting, and hence, we need to conserve the nature to protect plants, animals, and microbes. As you know, plants are the producers and animals are the consumers. So, obviously, there is a linkage between plants and animals in nature. Again, microbes are microorganisms acting as living entities available in nature and hit the target for their action. The actions they play are both positive and negative. We can see plants and animals of big kind through naked eye and part of it through microscope. But in nature, microbes are so tiny that cannot be vividly visible through the microscope in general. Sometimes, to see these organisms, we need high resonance ultramicroscope. The tiny microorganisms can be seen in mass if they form colony, means a great biomass after their growth over time. We always see molds on breads and roti, even on chapatis, fruits, when they are affected by fungi or bacteria and show color and odor change due to their interactions. On wood and on cloth, even on skin, we see many microbes that cause serious lesions, even rough texture. So, we are very much acquainted with microbes, but the types or kinds are unknown to us. In general, microbes can be used to produce many products and used as biofertilizers. Bacteria, fungi, and various bacteriophytes are used to act as biofertilizers. So we need to study these microbes to produce specific type of biofertilizers. Antibiotics, cosmetics, steroids, and several byproducts are produced by many bacteria and fungi. Day to day, the number of beneficial microorganisms used throughout the globe is increasing. So, scientists trying to discover the use of different strains and types for their action to make them congenial to us. In different fields, gene pools and the molecular markers are used widely to make the biotechnological tools for drug discovery, even to make hybrids and hybrids. In general, from plants to animals and from animals to microbes, a good chain of interactions always working silently to make the ecosystem sound and healthy. Our role is to study and research on this system to develop new components and to keep quite stable ecosystem for our future generations. Keeping this view in mind, today's program is very fit. Many experts, presenters, researchers, scholars, students, even contributors present in this August gathering. I hope the discussion will be fruitful in presence of the galaxy of workers for the same. I again convey my sincere thanks and gratitude to the officer in charge, GGDC Lalgore, and all members of the organizing committee, even all teachers and external guests for the presence to this conference to make it grand success. Thanks to all. Thanks again. Over to you, sir.
Thank you, madam. Now the time is for our very very dynamic sir Dyopan Mitra sir. Sir is always with us. We visited many places. Sir is from Department of English, but she is always very enthusiastic to study different types of organisms, even ecosystems, because environmental studies under different teachers going on for UG students. So Sir is also guiding many students in various fields. So this dynamic teacher will say something about the seminar or conference. Now Dyopan Mitra sir, he is our convener, seminar webinar subcommittee, GGDC Lalgarh, Jhargram. Sir, over to you.
Thank you, sir. Thank you. We are really beholden to Dr. Debabrata Das, Department of Botany, GGDC Lalgarh. We are also beholden to Dr. Pompi Ghosh from Seva Bharati Mahavidyalaya for joining us. Dear participant, distinguished guests, esteemed colleagues, it is with great pleasure and anticipation that I welcome you all to this conference on interdisciplinary approach on plant animal science and microbes, organized by the Department of Botany, GGDC Lalgarh, Jhargram. We are delighted to host this event on the 20th and 21st of December 2024 in a blended mode that allows us to connect both in person and virtually, accommodating participants from diverse locations. As we gather here, we are reminded of the interconnectedness of life and the importance of interdisciplinary collaboration in advancing our understanding of the natural world. This conference serves as a platform for scholars, researchers, practitioners from various fields to come together, share their insights, and foster innovative approaches that transcend the traditional boundaries. Yesterday, we have also seen a galaxy of performers who have enlightened us on different themes, relevant themes. And throughout this conference, we will explore a wide range of topics that highlight the intricate relationships between plants, animals, and microbes. Our discussion will also encompass cutting edge research, technological advancements, and the implications of these interactions on ecosystems and human society. We are privileged to have an impressive lineup of speakers who will enlighten us with their deliberations, will share their expertise, and inspire us to find their kind of new findings. We hope that this conference will not only deepen our understanding but also spark new ideas and collaborations that will contribute to the advancement of science and the betterment of our world. Let us seize this opportunity to engage in meaningful dialogue and build lasting connections. Thank you once again for joining us, and we really look forward to an enriching and productive conference experience. Thank you, everyone. Uh, Dr. Deobrata Das, sir, over to you.
Thank you,
Thank
sir.
you, sir.
Thank you.
Thank you.
Sari
Sari
Jolai
Jolai
Vidas.
Vidas.
So
So
now
now
we
we
are
are
moving to
moving to
the
the
uh,
uh,
special
special
talk
talk
part.
part.
So
So
here
here the
introduction
introduction
part
part
already
already
mentioned
mentioned
earlier.
earlier.
Now
Now
directly I
directly
just
I just
introduce
introduce
Profullo
Profullo
sir,
sir,
Profullo
Profullo
sir,
sir, Profullo Dr.
Profullo
Profullo
Sedi,
Sedi,
sir
sir
is
is
Associate
Associate
Professor,
Professor,
Department of
Department of
Microbiology,
Microbiology,
MAE
MAE
Abasayab
Abasayab
Gadwari
Gadwari
College, Pune,
College, Pune,
Maharashtra.
Maharashtra.
Sir,
Sir,
requesting you
requesting you to
to present
present
your
your
PPT
PPT
slides and
slides and
present here.
present here.
Now
Now
the
the
platform is
platform
yours.
is yours.
Over to you,
Over
sir.
to you, sir.
Thank you, Dr. Das.
Thank you, sir.
Thank you,
Thank
sir.
you.
Thank you.
Okay, I'll share the screen now, and from that point onwards, we can start discussing.
तुम्ही कोणाला स्क्रीनशॉट घ्यायची आहे
Is it visible to everybody?
Yes, sir. Yeah.
Okay, great. So, okay, okay. To begin with, my sincere appreciation and acknowledgement to all the organizers for their efforts in organizing such an interesting conference. And um, the important aspect of this is being a dual mode, online and offline, so that people who can't be physically present over there are still part of this conference and get to learn new things. So this is the best part of the conference that I can think of. And the second most important thing is the networking that allows to interact on an interdisciplinary platform where people are from a diverse background, though from the life sciences background. It's definitely a diverse background, people working in the field of microbiology, botany, developmental biology, or even zoology, aquacultures. All of them are on the same platform, and will have an opportunity to share our ideas and listen to the innovations that you are carrying. So, with this, let's start discussing the topic that I'm supposed to present in front of all of you. Uh, in the beginning itself, we heard about the sustainability and how people working in the field of life sciences can contribute to sustainable development goals, the global goals established by UN. We have these 17 goals in front of us. And the work that I'll be discussing in front of you today majoritarily covers ninth, 11th, and 13th point. So the ninth being industry, innovation, and infrastructure, 11th being sustainable cities and communities, and 13 being the climate action. So how microbes can help in all these three aspects will be discussed now. So the title of my presentation is exploring role of microbes in self-healing concrete, revolutionary approach. Okay. So I belong to state of Maharashtra. I work in Pune city. Our organization is affiliated to Savitribai Phule Pune University, formerly known as University of Pune. Uh, so the outline for the today's talk is in front of you. So I'll be explaining the topic through an introductory slides. Then I'll discuss about the hypothesis of our work, then the objectives of particular studies, the methodology that we followed, and ultimately I'll discuss the outcomes and the references. Uh, so to start with, when I'm talking about sustainable cities, infrastructure development, or building communities, and even climate change, one of the aspect in all three of them, which is common, is concrete. Uh, each one of us know concrete, have seen concrete, using concrete for a number of things. As a matter of fact, right now, the very place where we are sitting and listening to this particular talk is made up of concrete. If I'm not mistaken, hopefully nobody is on vacation in tents in forests or something, but the cozy home or the warm laboratories, offices that we make use of course, are built using concrete. So essentially, the composition of this particular component is nothing but cement, water, and aggregates, or various different components such as gravels or fly ash. Different aggregates are used, and combination of this based on the composition of or the proportion of cement and various different aggregates is the concrete. So this particular concrete is very strong, durable, and versatile in nature. The very base of the industrial civilization, the modern civilization, which we are part of, and the very reason of aiming sustainable goals, because this particular concrete also led to various different issues associated with it. If you talk about history of concrete, Romans are credited with the developing of this particular technology of concrete and building huge structures, huge infrastructure, as well as huge structures, bigger, larger, taller structures with durability and strength. Um, the current scenario for the global demand is to the tune of $410 billion, and within next 3 to 4 years, it is expected that this rate will increase, and the demand will reach to the tune of 515 to 12 billion US dollars, right? So as far as this component is concerned, this, though it's a global demand that we are talking about, it's not only the countries which are developed, or European or American continent. It's basically the continents wherein various countries are in the phase of developing or underdeveloped. So there the demand is pretty high as well. So Asia Pacific region, to which we belong, has the largest possible market as well for concrete. And essentially, when you talk about concrete, there is an environmental impact of concrete production. And that impact is on soil, air, water, and as well as health, human health, animal health, plant health. So collectively, when we talk about this, okay, so it's because of various different components that are part of this concrete. So cement manufacturing, or even the transportation of raw material or the finished product towards certain endpoints, right, that lead to various different impacts on the micro environment, right? So globally, that's the scenario. It's not only in developing countries, or it's not restricted only in India, right? Everybody working in this particular industry, right, is exposed to cement, is exposed to concrete, is exposed to hazards. What kind of hazards? The high concentration of various different metal or various different ions uh, is an issue. So your soil quality, the fertility of that particular soil, that's being challenged. The impact of air is being right, very high, very tremendous, considering the right amount of carbon monoxide or sulfur dioxide or nitrogen oxides or even dust particles that are being right formed or raised because of activities related to cement or concrete formation. So there is an impact on air as well. Of course, there is an impact on water and human health. So collectively, if you talk about this, though concrete is an important integral part of our day-to-day life, right, formation of that concrete poses various different issues. And the major issue, the most important issue, is with respect to environmental impact on concrete as well. We talked about impact of concrete on environment, concrete formation, production, right, and usage on the environment. However, there is a reverse relation as well. And what is that reverse relation? Environmental impact on concrete as well. And when we have this kind of environmental impact on concrete, the slide itself talks about it, right? We might have experienced these, right, wherever we work or our residents, right? So depending on the tenure for which we are using that particular place, at times even the brand new places, recently constructed places, will pose such kind of threats. So the environmental impact that we observe on the concrete, right, is in terms of cracks, right? Majority of them are either hairline cracks or are categorized as shrinkage or settlements or structural cracks. And as the size keeps on increasing, there is a threat associated with it, right? The hairline or shrinkage level cracks are still right acceptable or better in terms of right working condition, but when you have a settlement or structural right cracks, then they pose various different threats and issues. So classically speaking, most civil engineers, people working in infrastructure development sector, can categorize such cracks into different 17 different categories, right? And what's the reason behind formation of these cracks? The environmental impact, of course, but that environmental impact is categorized through physical, chemical, thermal, plastic, and structural impact, right? So the physical, such as drying or creasing or shrinkage kind of thing, a chemical such as alkali aggregates or steel corrosion or carbonation, or thermal, such as early thermal contraction or freeze thaw cycles. So these seasonal variations also lead to freeze thaw cycle, also lead to thermal contractions, and they may lead to formation of cracks. Okay, so once you start with a brand new structure, and very soon if it is being corroded, if it is being degraded, if it is being right converted into rubbish or converted into a rubble of the material, then of course there is a necessity of fresh concrete. That means again the same cycle wherein industrial production of concrete will increase, then its impact on the environment will also increase. So that cycle, that vicious cycle, keeps on continuing because of deterioration of concrete structures. So what we thought, right, to work on this particular aspect. So if you talk about the durability of concrete, there are various different issues associated with durability. Of course, everybody who works with these kind of raw materials wish to create the most durable, right, piece of concrete, right, piece of infrastructure, a building, a wall, or whatever it is, right, a beam, pillar, or even the fences, ceiling, right. So the issue is durability of the existing structure is not to the expected end. We talked about Roman contribution in formation of concrete, right? Their structures are standing there for centuries together. But can you claim the same for existing buildings, existing infrastructure that you have established using the current trends, current scenarios in the concrete manufacturing? And I feel really sorry to say this. We don't have, right, that assurance that, okay, these structures will exist for those many years, right? Our acceptance for existence of these structures is roughly 50 years or so. In exceptional cases, it will increase below or beyond 50 years or so, right? So that's the reason, that's the main focus point where reuse, maintenance, and repair also need, right, additional amount of concrete being manufactured, and it will have its effect on global environmental changes. So the reason is additional requirement to repair and to maintain. So, is it possible to come up with alternative, sustainable means or methods for repair and maintenance of these kind of structures? So, yes, definitely it is possible. There are two different approaches. First, replace concrete with sustainable concrete, right? So, wherein you have option of recycled material. These days, our honorable minister, uh, uh, Mr. Nitin Gadkari ji, keeps talking about using recycled material for construction of highways. So they make use of plastic, rubber, and whatnot, right, as a recycled material. So it helped them to keep it more sustainable, and the cost is also reduced, right? So the same principle can be applied for concrete as well. Then we have other options such as organic aggregates, so fibrous material. The people have used various different fibrous material, including fungal mycelia as well, right, to generate the sustainable concrete for durable concrete. Apart from that, people have used synthetic fibers as well, right? So this is one approach, replacing concrete. But then, considering the demand of concrete, the amount of concrete required, amount of strong durable material required, it's practically impossible to replace a concrete, right? So now the other option, which is practically deemed suitable, practically feels suitable, is using reinforced concrete. That means you change the repair or maintenance strategies and make sure you need minimal cement, minimal concrete formulations, right? And with that, you can have a durable, right, concrete formation. And this particular site, right, talks about the same. Here, under the category of reinforced concrete, you have a couple of images, right, from 0 day, 1 week, 2 week to 3 week, wherein you can see that self healing of that particular crack, right, can be observed. Now, we attribute healing as a character, as a quality, right, to all life forms or living beings. You have a cut, maybe a paper cut or a metal scissor cut, blade cut, or maybe a wound, right? No matter how deep it is, or no matter how superficial it is, in case of plants, animals, and human beings alike, right? These life forms, they have self-healing capacity, right? So these wounds, these cuts, they will heal on their own. Of course, medication will expedite the process, but most of them, they will heal on their own over a time period. So, concrete being a non-living entity, of course, won't have this tendency on its own, because self-healing is attributed as a character to only living entities, life forms. Concrete is a non-living entity. But still, you can see over a period of 3 weeks, you can see that crack has been self-healed. So how is it possible? So this is where our work comes into picture, where we are trying to work with self-healing bioconcretes, right? So what is a bioconcrete? Essentially, bioconcrete is a mix where you use cement, you use water, you use other materials such as gravel, fly ash, fibers, right? Whatever aggregates that you are using to form concrete, but additionally you use microorganisms as well. So you add certain microorganisms to this particular system and make use of them. Okay, so when we talk about this microbially induced system, okay, so this is what a bioconcrete is. So this particular slide over here talks about the same. You have a concrete composition, right? Same as the classical concrete. The only difference here is you're adding microbes, you're adding bacteria to this particular mixture before it is being casted, before it is being cured, right? You have that concrete casted in a pillar or beams or maybe ceilings, right? So as a flooring, whatever it is, right? Before you cast it, you add microorganisms and then you cast it. Okay, and what kind of microorganism? Of course, very specific category of microorganism which are capable of forming crystallization, capable of biomineralizing, right? Various different entities. So these microbes should have a very peculiar character, a specific character to say microbially induced calcium carbonate precipitation should be, right, associated with these particular organisms. So there are mechanisms, there are categories, right, through which microbes can achieve this calcium carbonate precipitation. And why you need calcium carbonate precipitation? For self healing, right? So this is what is happening over here. The crack, the moment the crack is formed, the microorganisms which are present in the concrete, right, they start working, right, in that micro environment, utilize nutrients that are already there in that particular concrete or that particular crack, utilize air, oxygen from the micro environment, and start, right, precipitating calcium carbonate. And that calcium carbonate is acting as a cementing material, is acting to close that particular crack, to fill that particular crack, to seize that particular crack. And this is how your bioconcrete works. Okay, so these are the mechanisms through which a particular microorganism may precipitate calcium carbonate. And these mechanisms are urea hydrolysis, ammonification of amino acids, denitrification, and oxidation of organic compounds. There are certain other methods as well through which microbes can, right, precipitate carbon, right? So if we can work with such organism, you can develop your own self-healing concrete. So that was our idea. And with that idea, right, what we thought, once we have that particular bioconcrete, once we have that particular self-healing concrete, will it be useful? Of course it will be, right? So bioconcrete can have a higher compressive or split tensile or flexural strength than the conventional concrete. So if you are having more compressive strength, more tensile strength, more, right, flexural strength, what does it mean? Your weathering, environmental effect on this particular concrete will be minimal. And not to forget, once you have such kind of, right, structures, right, or structures that are there, of course their lifespan will be much higher than 50 years or so. right? One such study found that bioconcrete with 1% biocement, right, had a 15.34% increase in compressive strength and 15.17% in structural strength after 28 days of curing. Okay, so this is the most important benefit that you are getting: strength. Another important benefit that you are getting is self healing, right? The moment you have effect, environmental effect or any other effect on that particular structure, on that particular concrete, and if it is leading to any sort formation of cracks, the self healing property of that particular bioconcrete uh, plays a pivotal role, and that crack will be self healed immediately, quickly. And when I say immediately, it's not within the fraction of seconds or within minutes. Of course, you need some time, but right, rather than deteriorating further, rather than eroding further, rather than increasing from hairline crack to the structural crack, of course, it can be cured, it can be created immediately, right? So that's the important aspect. Apart from that, recyclability and its use makes a huge difference with respect to sustainability and sustainable goals, right? So these are the most important advantages, right, of bioconcrete. Now, when you talk about introducing organism in something, especially the walls, right, that you have, or the pillars, the ceiling that you have, of course, it will have its impact on health and safety as well. So, right, this particular notion that when you are using microorganism, when you are introducing microorganism in concrete, and you are living in that particular structure, you're spending your days, right, years in that particular structure, is it safe? Of course it is safe. Why? Because the organism, the type of organism that you're using, is a non-pathogenic in nature. And in our subsequent slides we will discuss about this, how to check that as well. Okay, so with reduced carbon emission, with reduced maintenance, with increased durability, with increased strength, and most importantly with increased self-healing capacity, bioconcrete is, of course, the right talk of the town with respect to sustainability development, right? So based on that, what was our hypothesis for the study, the research work that we undertook? The hypothesis that we had was diverse bacteria population under varied conditions are capable of inducing calcium carbonate precipitation that ultimately improves structural and durability properties of the concrete. So our aim was to work with diverse bacterial population, check their capability, whether they are an ideal candidate to form a bioconcrete. So our objectives were very simple, pretty much simple, just two objectives, one of which talks about isolation and screening of bacteria having potential calcite production, right? So calcite is the calcium carbonate precipitation, and the second objective was identification of bacterial isolates showing optimal calcite production and characterization of calcite crystals. So it was a simple, right, exercise that our students were involved in. So one of my PhD student, she worked on this particular aspect, so it's her contribution. So we collected our samples from two different districts in Maharashtra, one of which was Anjaneri mountain. Um, there is a correlation of Ramayana with this Anjaneri mountain as well. And the other place was in Satara district, another place wherein we have caves, various different caves, which were pretty old, almost from the periods of Buddha. So those caves were our sampling site. Here we have our sample containers as well. So we collected various different samples, the speleochem samples, stone samples, agricultural soil or stone scrapings, and weathered ruins that were there. So we collected samples from all of them, and those were used for isolating various different organisms. So this was our protocol. This was our experiment for enrichment and isolation, wherein we suspended all these samples, right, in a specific medium known as CCP medium. This is a calcium carbonate precipitation medium. Along with that, we also use B4 medium, wherein calcium carbonate precipitated calcium is provided into medium in the form of various different systems. So apart from that, the incubation period that we used was pretty much prolonged rather than over incubation of 24-hour incubation, which is most common in case of most of the microorganisms, if not for all. Our aim was to enhance calcium precipitation, so enrichment was done for 10 days. Subsequently, post enrichment, so if you go through these images wherein we have flasks of various different media. So this is your CCP medium. This is how it looks, and the B4 medium. This is how it looks. So the A flask is our control, uninoculated control. B is the one where we inoculated our samples. So after incubation, what we observe, right? So this is, and these two images, this is basically a zoom in for the tilted flask. So these two images are of a tilted flask wherein we observed precipitation, right, at the bottom on the edges, inner surface of the particular flask, right? So this particular precipitate is of calcium carbonate. So over a period of 10 days, so much of calcium carbonate precipitate can be formed, right, by these microorganisms. So of course, these microorganisms which are present in that particular sample are no doubt the most ideal candidate for this particular work, right? So subsequently we went for isolation, wherein serially diluted samples were used, and now instead of B4 and CCP broth medium, we have used B4 and CCP agar medium for isolation. So over a period of 4 days post incubation, we could isolate 36 different morphotypes on B4 agar and 58 different morphotypes on CCP agar. Morphotype is ideally a distinct colony, right? So based on that, we could isolate those many bacterial isolates. So this is how the isolation progressed for various different samples. So these are certain plates wherein isolated bacterial colonies are depicted. So once we had those 94 different isolates from B4 and CCP agar, we started our screening work.
मसाला
So the
चाहिए
primary
मसाला
screening
चाहिए
for calcium carbonate production, calcium carbonate precipitate, was by using the stereo microscope and fluorescence microscope, right? So simply we observed the colony of these particular organisms, right? Isolated colonies of these particular organisms growing on B4 medium, growing on CCP medium, right? Containing calcium source, right? Whether these organisms, whether these colonies are crystallizing something, right? If they are forming crystal, that is an indication that a precipitate is being formed, right? And subsequently, the another step for screening was, right, calcium carbonate production by the selected isolate was confirmed through acid freeze test, wherein 10%, right, hydrochloric acid HCl was used. So these are the observations that we, right, um, had for the microscopic systems. The first particular image, these two images are of fluorescence microscopy, wherein you can see the crystals are being formed, right? So these crystals are of calcium carbonate. On similar lines, when we use stereo microscope, right, we observed crystals, right, beneath,
मोबाइल
around,
में चार्ज
and
मारना है मोबाइल तो
within
मोबाइल
the particular colony. Okay, not only that, when, right, we zoomed in, we could also see the structure, the crystalline structure of this precipitate, calcium carbonate precipitate. So this was our primary screening system. This was our primary screening test where we could confirm that yes, organism is capable of, right, precipitating calcium carbonate. And this is the observation of acid phase test, wherein, right, after the incubation, we precipitated our entire calcium carbonate by the microbially induced precipitation. We removed the medium, we removed the broth, we removed the biomass, and, right, after the separation, whatever precipitate was adhered to the inner walls of the container, we could see fizz being generated in the acid phase test. So this is because of, right, release of gases. So calcium carbonate plus HCl, there will be release of CO2. So that fizz is generated. This is another affirmation that gives the confirmation that a calcium carbonate precipitate is there. So once we had this, now we started characterizing these organisms that we shortlisted. So based on the microscopic observation and acid fit test, we shortlisted 27 different isolates, and we started working with those 27 different isolates out of 94 isolates that we had from various different samples. Right now, if right, the microbiologists are studying this particular part, of course they have their own ways of deciphering the questions, but any microbiologist will think why certain basic preliminary tests such as gram staining and endospore staining is being discussed as a part of this particular presentation. There is a very important reason associated with it. We will talk about this, but then let me just tell you that these were the two criterias under which the characterization of these 27 isolates was done. The first criteria was gram nature and capability of formation of endospores, and the second character was urease, right, formation capability. Okay, so to talk with gram staining characters, if you can check this particular table, the observation for all 27 isolates, right, right, where with respect to gram positive organism, right, none of them were gram negative, and this is a good thing. Why good thing? Because as I said in the beginning itself during discussion for introduction, safety is an important aspect, right? So gram positive pathogenic cultures are very few. Of course there are, but very few. Instead, gram negative organisms, right, tend to be more pathogenic. So those known pathogens, right, the majority chunk of those are gram negative in nature. So organisms being gram positive is an important aspect. So this is one step towards the safety, right? The second part, whether the organism is capable of forming endospore or not, right? It was observed that organisms are capable of forming endospores. What is the significance of formation of endospore? In bioconcrete, as I said, if you are adding microorganism, right, and curing that particular concrete for days and months and using that concrete for years together, it is not sure that the crack will be formed immediately. Maybe after 2 years, 3 years, 4 years, 10 years, it will be formed. If the organism is capable of forming spores, it can form a spore and remain there for that much long period, right? So once it is exposed to favorable conditions, once the crack is formed, what will happen? That crack will be exposed to microenvironment. So that crack will be exposed to air, that crack will be exposed to dust, dirt particle, and the nutrients that are there with dust and dirt particle, right? So if the spore former sense that favorable condition, nutrient availability, oxygen availability, air availability, of course, it will, right, right, convert itself into a vegetative form, and it will start growing, multiplying. And as it grows and multiplies, it will, right, because of its characteristic tendency, will start precipitating calcium carbonate as well. The tendency that we are looking for, that's the important aspect that we want. Okay, so this is how it starts and works. Then subsequently we also studied urease test. Why urease test? Because in the beginning itself we talked about there are four prominent mechanisms through which calcium carbonate precipitate is induced by microorganism, one of which is through urea hydrolysis. Urea hydrolysis is possible only if organism is capable of urease formation. Okay, that's the reason we check urease as well. So subsequently, when we started this particular first objective, our aim was to isolate certain organism with specific qualities, right? So to summarize our first objective and its outcomes, right? Total of 94 morphotypes were obtained from 11 different samples, right? Right, all the isolates were microscopically screened for a crystal formation, and we found that 27 are the prominent one, and those 27 were then subsequently, right, analyzed, screened through urease test, gram staining, and spore forming, right? So after these tests, we observed that 17 isolates are capable of forming urease, right? So those 17 were then subsequently selected, and the next step was secondary screening based on quantification, right? What kind of quantification? Quantitatively, we studied how much amount of calcium carbonate can be precipitated using a specific organism. Of course, the method was very crude. We used gravimetric method, and these were our results. So all of them are capable of, right, precipitating calcium carbonate. However, when we compare that amount in terms of gram per liter, right, there is a variation. At some point it is more, at some point it is less. So considering these variation, then we shortlisted few more, right, right, then cross checked, and majority of those, if you can see, are more than 5 gram per liter. So this was our secondary screening. So these shortlisted ones, right, the 8 different bacteria we shortlisted. Finally, we started working with them and started characterizing the calcium carbonate crystals that are formed by these organisms. Okay, so of course, I'll be discussing data of certain representative isolates. So this is with respect to scanning electron micrograph of the calcium carbonate precipitates, right, obtained from 4CS6 isolate and 15CS5 isolate. Okay, so you can see their crystalline appearance, the precipitate, right? And there are various different ways through which these crystals are being formed, and various different structures of which these crystals are being formed. Okay, so we also analyzed these crystals, right, through FTIR. So this is the top one, is the standard FTIR spectrum of the calcite precipitate, right, calcium carbonate precipitate. And if you can compare the same with the following two images, the second image is of FTIR spectrum of 15S S9 precipitate, and the third one is of 4CS6 precipitate. So this signature spectrum of the calcite precipitate can be observed in the samples that we generated through microbial induced systems, right? So this also confirms, the FTIR studies confirms that there exist these particular properties with these particular organisms. Then we moved towards identification of these bacterial isolates. So 16S rRNA gene sequencing was done, and it was observed, it was found that 4CS6 is actually a Penibacillus fonticula isolate, and the 15CS9 is Porosarcina luteola, right? So both these are capable of precipitating. Now, Penibacillus or majority of Bacillus strains are reported for having these kind of properties. So subsequently, when we worked on this, the FTIR and EDX analysis or the scanning electron micrograph analysis suggests that yes, certain organisms are there and they can effectively form calcite, and these can be effectively used in self-healing concretes as well, right? So to confirm this, subsequently, right, we started studying other mechanisms as well, because as I said, there are multiple mechanisms through which microbially induced calcite formation is observed. We studied initially only one mechanism, urea hydrolysis, but it is not so that only urea hydrolysis is there in the particular isolates, right? So all the 26 isolates that were those were capable of forming spores, right? We started screening those for various different tests. The reason behind this is, if you are using urea as the source, right, as a nutrient, then the organism is capable of urea hydrolysis, will be the useful isolate for bioconcrete. However, if you are not using urea, then the organism is useless. So we need to have multiple mechanism in the same organism so that it becomes a important candidate. And then once we performed all the screening, right, assays, what we did, we studied whether there is an isolate which shows all three mechanisms, right, such as oxidation of organic substrates, nitrate reduction, or urea hydrolysis, right? So when we went for such kind of analysis, such kind of test, of course, what we observed was such isolate do not exist, but certain organisms are capable of, right, working with two different mechanisms. So that was an advantage. So these are the results of our tests that we performed, right? Urea hydrolysis and nitrate oxidation and, right, other aspects. So once we performed these tests, right, we went for another additional test that was important, that was crucial, and that test was acid production. Now, what is the relevance of this particular test, acid production, in bioconcrete formation? The relevance of this particular test, acid formation, acid production, is, right, acid itself is a causative agent, itself is responsible for biodeterioration, biodegradation, or damage of concrete. So majority of organism, as we know, are capable of producing acid. So if acid is being formed, what's the issue? The calcium carbonate precipitate will be solubilized by the organism itself. The acid will work on that precipitate. We don't want that to happen, and that's why we started looking for these particular aspect, whether the isolates that we are shortlisting are capable of producing acid. If yes, how much amount they are producing, right? And so for that, we performed this particular test wherein, right, we observed, right, whether the organism are capable of producing acid or not. And what we observed, the acid production is absent in particular isolation, right? So we don't see acid. So there is a relation of fermentative utilization and non-fermentative utilization of carbohydrates available for this particular organism, right? So basically there was no halo formation around the particular, right, plate that contains calcium carbonate. So organism growing on the medium, if produces acid, right, it will solubilize the calcium carbonate in the vicinity and will form a halo. Not only that, right, the organism growing on a medium containing pH indicator such as phenol red. Of course, the organism produces acid, the indicator will change its color. But in both these systems, wherein we have used calcium carbonate supplemented medium or phenol red supplemented medium, we haven't observed any formation of acid, so which is a good thing. But we didn't want to rely on only that aspect. We wanted to study, right, calcium carbonate solubilization as well by these particular isolates, and that is why we used Devizy Bruni medium to check the calcium carbonate precipitation solubilization studies. Herein we have these particular plates where we wanted to check whether, right, we have this particular tendency of an organism. Now all the organism except this one, right, here 4C56, right, S6 is the one which is giving halo, right? So there you can observe halo. What does that mean? That particular isolate is, right, forming a halo. That means it is producing slighter quantity of acid and is capable of solubilizing calcium carbonate. So if somebody asks me if this is an ideal candidate for bioconcrete, I'll say no, this is not an ideal candidate. Despite of having mechanism for calcium carbonate precipitation, it also harbors a mechanism to solubilize that precipitate. So we don't want to use that particular kind of organism, right? So this is the beauty of particular aspect where these organisms can be used in a bioconcrete formation, right? So our studies here suggest that there exist certain genera, certain species which can be effectively used as a candidate for bioconcrete formation, and we successfully could isolate at least 26 different morphotypes of such kind. And as we are identifying those organisms, we are getting to know that these belong to Bacillus sporosarcina or Penibacillus genera, right? So this is an important aspect. Okay, so if you compare it with the same aspect, again, I'll revisit my first slide, the global goals for sustainable development. I think microbes definitely can play important, significant role in almost all these aspects, all 17 of them, almost all 17 of them, right? Okay, but if not indirectly, directly in ninth, 11th, and 13th. They can definitely contribute. And the organism that we are working with are the most ideal coordinates for these kind of differences and these kind of changes that we expect under the sustainable development. Okay, so with this, I'll move to the reference section. So these are the references that were mentioned in the slide. So this
ডেটা
is
লিমিট রিচ করে
all
গেছে
from my side.
এটা করে দিতে হবে এটাকে ওয়াইফাই করে দিতে হবে।
Sir, hello, sir.
Yes, sir.
Hello. Can I ask a question?
Yes, sir, definitely.
থ্যাংক ইউ
Thank you,
স্যার
sir,
ফর
for
ইউর
your
নাইস
nice
প্রেজেন্টেশন
presentation.
থ্যাংক ইউ স্যার
Thank you, sir. Thank you.
Thank you.
ওই যে ওরা কোশ্চেন করছিলাম তোমার ভালো সাহাটা
Hello. Hello.
Yes, sir.
Sir, exactly as it is healing the crack, when the microorganisms start its work without any crack within the wall or concrete, they will not function.
See, I haven't presented the data here, but we also check whether they are aerobic, microaerophilic, or anaerobic in nature. Our assumption is within the concrete, inside the concrete, there will be anaerobic environment and microorganisms won't be able to grow over there. Another aspect, though it is porous in nature, the oxygen levels within the concrete, inside the concrete, will be minimal or sub-zero. However, the moment crack is formed, no matter how tiny it is, the moment crack is formed, air will be introduced inside. That means oxygen will be introduced inside. All our isolates, as they are spore former, they are microaerophilic as well, right? So
Yes.
with the minimal amount of oxygen that is present, they will start multiplying. They will be right viable then, and then they will start multiplying. They will start working on it. So that is the assumption.
Yes. Thank you, sir.
So no
So no
questions.
questions.
Thank you, sir.
Thank you,
Thank
sir.
you.
Thank you.
Thank you very much.
Next
Next
resource
resource
person
person
was
or
Dr. D.
Dr. D.
Thangamani,
Thangamani,
scientist,
scientist,
Institute of
Institute
Forest
for
Genetics and Tree
Tree
Breeding,
Breeding,
Coimbatore.
Coimbatore.
Madam,
Madam,
now your
now your
session.
session.
Over to you,
Over to you,
madam.
madam.
Good evening, everyone. Is my slide is visible, sir?
Yes, ma'am.
Good evening, everyone again. And my heartfelt thanks and applauses to Dr. Das sir and Bambi ma'am and their team of Department of Botany, GGDC, Lalgarh, Jhargram for this dedicated work to the Society of
Babu from
Science.
Kuchi Betar.
And I thank all the participants for their presence here. Let us start my talk. So, on characterization of genetic resources using molecular markers. Genetic resources, I mean anything that is having the hereditary material, microbes, plants, or animals, right? So everyone we know about the Charles Darwin. Charles Darwin, yeah, and he's an excellent natural scientist, and he roam around this world. He visited different countries and continents using his ship named as Beagle, and he has written a book, The Voyage of the Beagle, and another book is on the origin of species during the year 1831 to 1836, and he wrote about his theory and his experiences regarding the species and the changes and the variations he saw around this world as evolution by natural selection. So he has given a five main points how the variation could have happened in the nature, and he has abbreviated this one as variation, inheritance, selection, time, and adaptation. Okay, in his note, see in the left hand side, he has written in his scripts like I think it is a tree, its evolution tree. The variation comes in generations. He has written in his scripts. And later, Lama came and he said it is the variation that is inheritance has occurred. It is not. Um, it is not through evolution like that he has said, and we have a doubt whether variation still exists or in the gene level, or we still evolving. So the answer is yes, there is a change in the gene frequency in our DNA. So this is called as microevolution. And is there any macroevolution, that is the formation of new species likewise? And we can say it is no, right? So for the answer for are we still evolving, I said there is a chances, there is a evolution, microevolution in the gene level. So for example, one of the gene called CC45 gene, okay, this gene is responsible for carrying a resistant of HIV-1 infection. So this gene is totally absent in African population, but this gene is present in European and West African populations. Okay, so the scientists noted why this has happened, because then they found out, because this is due to the mutations that is originated in Eurasia. Okay, in those that continent, that areas, because of the mutation, this microevolution has happened. So this caused the this microevolution. So this happened because of the pandemic disease plague. Okay, so this type of microevolution, because of corona also we got this resistant, right? So this and all microevolution. So like this this variation is happening for several years, right? From the start, there are millions of years, the variations transfers from one generation to another generations, variation continues and we evolve, right? So we cannot argue whether it is two legs are good or four legs are bad, like that. As a scientist, we cannot no, we cannot argue, right? Everything is good because of its adaptation and because of its evaluation, right? So we always welcome the technologies. Okay, so evolution is always good, right? So now from the Stone Age period we evolved up to AI period, so evolution is always good. So there is a lot of diversity because of evolution. And where it has come from? So it is come from nature, right? From the biodiversity, from the nature, from the biodiversity of microbes, plants and animals. And this variation has comes and the evolution takes place. So the reason for something, there is more similar than others, right? So what it looks similar but it is not the similar one, because of its heredity, right? So because of the hereditary material modification, how it was changed. So that's descent with modification from a common ancestor. So what is the mechanism behind is the variation. So variation is the important part plays in the genetic resources. So because of these variations, right, we are getting a very novel characters. So the similar species passport data was cataloged by Linnaeus. So he has given a classification. So something are more similar than others, but it is not similar, right? So he cataloged into a different orders, different families, right? So genera, etc. So after that Smith came, he has also classified the kingdom into a prokaryote, eukaryote, fungi, animal, protozoa, etc. But still there is a variations, right? So now we are following APG classifications, right? And using Gamble volume we are finding the different families, different species, genera, etc. Okay, so these variations, this genetic variation within the species among the population, how it happens. So this phenotypic variations between the species, red color, blue color, different type of banana, different type of brinjal, different type of tomato, all this variations species reflect is genetic differences between the species. So this genetic variation across the species is the origin of genetic variations, right? So where it is origin, it is ultimate source is mutation. Because of the mutation in within a species, this sort of differences and variations occurs. Okay, so our goal is genetic. What is the basic genetic mechanism that determines the traits expressed by the individuals in a populations? So for that purpose we are doing the characterization. Characterization refers to the passport data of a particular organism. It can be a plant or a tree or an organism. So to get the full description or passport data of a particular organism, we have to do the morphological characterization, then we have to do biochemical characterization, then we have to do molecular characterization. Okay, so this will show us how they vary from each other, right? So genetic variation it is very much important, and it is the process of evolution, and due to change in the genetic makeup of an organism, this variation occurs, right? And this variation passes from one generation to next generation. So it is also occurs due to environment, but we have to confirm it is due to edaphic factor or environmental factor or due to genetic changes, right? So there are three different components of diversity. One is genetic diversity, another is species diversity, and ecological diversity. Okay, so this genetic diversity, for example, in India, especially in the eastern India, there are northeast India and more than 50,000 genetically different strains of rices are available. Okay, so similarly more than 1,000 varieties of mangoes are available. And when we come to the species diversity in western Ghat areas, it is greater with more than 500 different amphibian species, frog species. And similarly, coming to the ecological diversity, different type of desert, rainforest, mangroves, coral reef, wetland, estuaries, so on. So there is a great diversity, right? So the variation within the genetic resources of microbes or plant or animals is very much important, and it is giving an enormous economic value to this society. It gives food, it gives fuel, it gives timber, it gives medicines and fruits and non-timber forest products, and also it does regulatory services like carbon sequestrations, and it prevents soil erosion, and it augments the groundwater, and there's several industries and several companies are benefited, stakeholders are benefited out of it, right? So who found the variation within the species first, and who has played a major role? We can say that Dr. Norman Borlaug is the father of Green Revolution of America, and during the feminine period, during the year during 60s, 50s, and all, and he roamed around all the villages, and he found the dog variety with more grains and tall variety with disease resistant variety. He brought and he crossed, and he got a variety with the excellent grain production with the disease resistant variety. He has given this grain to all poor farmers, and the farmers got benefited in tons. In acre, they got in tons of grains, and because of that, the famine got demolished. So he got a Nobel Prize for that. Is Green Revolution during the year 1970, right? So this is he found the variant from the nature. Okay, because of the mutation, and he could find the variant, the protective variant from the nature. And similarly, in our India, Dr. M. Swaminathan, he's the father of Green Revolution in India. He also worked along with Borlaug, and he roamed around the northeastern parts, and he has also collected the different variety of rice and wheat, short and tall. He also did the crossing and everything, and he also given a wonderful varieties to the farmers, and our family also went. Okay, so poor farmers have benefited excellently, and we got very excellent new varieties because of its variation. So how to do this? How to find out this variation in the plants or animals or microbe? First, we need some basic tools, right? We need some basic tools for identifying the colors or whatever it is. Then we have to feel it. We have to feel, touch and feel what is the what is there or what is not there. This is one of the well-known plant, that is teak. Everyone knows Tectona grandis, the timber tree. So this teak plant, we have to feel, can feel, touch and feel the presence or absence of the anthocyanin content.
resource person.
Then the
Thank you,
variation
madam.
in this, I'm showing
Very
some of
nice
the
talk.
examples, okay? Some of the
Next
examples
research
how
person, Dr.
we can do the morphology in microbes also, by growth pattern, by the secretion of the enzymes. So all those things we are noting down for the finding the leaf morphology. Similarly for the animals also same thing. So here see the variations in the morphology. Some are delta, some are coordinate. So all these things we have note down, and see the variation in the leaf petiole, the length. Some are sessile, some it is present. You have to note down everything, and see the tip of the tip shape of the leaves. Some are accused, some are acute, right? And see the adduction and abduction, that is upper and lower portions of the leaf. Some are very shiny, glabrous, some are leathery. Okay, so this type of variation also we have to note down morphologically, and see the trichomes, that is hairs present on the leaf, whether it is in a low or absent, it is high or medium, and we have to measure the trichomes structures also, okay, for positive data. And we have to record the bark type, stem type, what type it is, whether it is a scaly or a smoothie or fissured, and what color it is. Everything we have to record. And similarly in eucalyptus we have record. See the reticulum bark, how the peel, how it peels, and how they vary the bark. This also we have to note down, and see the arrangement of leaf, whether it is opposite or a decussate or a ternate. All those things also we have to note it down. And same thing, this petiole length and leaf morphology, this one also we have to record. And see the variation in the branches, right? All are look similar, but in place to place it differs. And the branching pattern also important characters in characterization. So we have to record everything. And similarly the shape or the crown shape of any tree or plant, and whether it is lanceolate, whether it is a conical or a columnar, this one also we have to record. And as I told you, the branching pattern and the branch and the angles also very important, right? And the flowers, all are teak plant flowers only, but see the colors, from some is pink, some is yellow, some is white color. And similarly the petal numbers and the anther numbers, this also varies. So we have to keenly watch and have to record the variations. So when the whenever the branch or leaf of falls down, it makes scar on the tree or plant. So this is also a very important trait. It's a very stable characters for the plant or this one. So we have to record that one also. So likewise we recorded the morphological characterization for rose root. So using image analysis software we recorded the leaflets, first leaf, second leaf, third leaf, and fourth leaf, all this one, the quantitative characters we have recorded, and we correlated with all other species what we have explored across India. And after this we have to do the biochemical variations also, to find out the biochemical variation, whether any changes in the primary metabolites, that is carbohydrate, lipid, and protein, and in secondary metabolites, in tannin, all in phenols, in terpenoids, in nitrogen containing compound, in sulfur containing compounds, what type of variation it is present or not. So qualitatively and quantitatively we have to see the variations. Then we have to go for the molecular characterization. So this molecular characterization is very much important. It cannot be visualized, and only after isolating the DNA and with the help of the marker we could do this one. So everyone knows about the nucleus, and nucleus contains the hereditary material, that is chromosomes. It is a double helical structure, everyone knows, right? So this genetic variations can be seen in the genes, the chromosomes, and the genes, and in the function of the proteins. Okay, so it is a very much important. Okay, in this purine and pyrimidine, that is the codons of adenine, thymine, and guanine cytosine pairs. If any single changes, in 1% only, any single change in the guanine to cytosine in one place, it changes into C to G, then SNP site it is, that is single nucleotide polymorphism has happened. Okay, so at least in 1% of the population if it occurs, means there is a variant. So because of this this change. This change, we can get a very sweet mango, very different aroma of resin, or different variety, new variety, anything we can get because of this small change. This is the variations, right? So how to find the variations? So there are two different type of mutations. One is chromosomal mutation, another is gene mutations. Okay, so it are the changes the genetic material. So these are all the two basic types of mutations. So chromosomal mutations produce changes in the whole chromosome, whereas the gene mutations that produce the changes in a single gene, right? So because of this mutation, what type of changes happens in the chromosome? Because there is a change in the chromosome number, it may happen, or changes in the chromosome structure. So what are the four type of chromosomal mutations? That is deletion, or duplication, or inversion, or a translocation. Okay, so these four can happen. So this cause mutation. So what is significant of this mutation? Most of them are no little, no effect. Some cause very change in the genetic activity or protein cycle. In that case, we can see the stunted growth, or any disease. They will be susceptible to many diseases, like it will lose its metabolism. Such a kind we can see. So in and some new species also can be evolved. Okay, so this is the source of genetic variant species. But polyploidy condition also come. That is extra set of chromosome also come. So this plants, we can see it's a robust growth. Okay, in human also, animals also it has happened, and in microbes also same thing, and in trees and plants also same thing. Polyploidy conditions can come. Okay, so how to find out? Okay, there are two types of markers. One is dominant marker and codominant. This DNA markers, that is markers, play important role to find out the mutations, that is variations in the plant. So this dominant markers, we cannot see the allelic variations, right? So allelic variations we cannot say. That is codominant individual loci. What is the exact translocation? We cannot see. But in codominant marker, it displays both homozygous as well as heterozygous state of the alleles, right? So we have always go to the heterozygous marker. Okay, so for your information, that homozygous means it is the same in everyone knows this Gregor Mendel's Punnett square, right? So it is a both are homozygous means it's GG, and it is two heterozygous means capital G and small g. One recessive means GG. So this each gene has two copies, which are called as alleles. This is called a G. So two alleles means for one G. So if both are coming from the same arms of chromosome, it is a dominant allele. It is from the same arm, can be a recessive allele, or it is a heterozygous. So this variations we could study only using the codominant markers. Okay, so the different type of markers we can choose based upon our applications. Okay, so RFLP is there, and polymerase chain. Because of this polymerase chain reaction, after the identification of tag DNA polymerase, there is a PCR engine, so it amplifies a DNA into a million copies. So using polymerase, tag DNA polymerase and PCR engine, there are different type of markers we are using to find out the genetic variation. That is RFID, ISR, intersimple sequence repeat, and amplitude fragment lipid polymorphism, and simple sequence repeats SSR, and SNP single nucleotide polymorphism, scar markers, ESV expressed sequence tags, etc. So there are n number of markers are available. So how this marker can be applied to find out the variations? Okay, so there are three type of DNA technologies. One is DNA barcoding, another is DNA fingerprinting, another one is DNA profiling. Right. So where and all these three technologies applied. So to distinguish between the species, you and me, the DNA barcoding is used. So distinguish between the individuals within a class, within an area, this DNA fingerprinting is, that is clones, we can say. Okay, this DNA fingerprinting is used. So distinguish between the populations, this DNA profiling and population genetic studies, this DNA profiling is used, right? So DNA marker is applied in various fields according to our application. So it can be used in DNA fingerprinting studies, diversity studies, marker assisted selection, genetic map, gene taking, and for F1 generation identifications, seed testing, etc., etc. Right. So if it is we are using for genotyping, we can typing. So which gene, it belongs to which genera, how the species has evolved, whether all the species comes under the same species, how they differ from each other, what is the genetic distance, so what is the variations to provenance level, region level, national level, country level, all those things we can study. Then population structure analysis, how the population is, how the variation is, all those things. Then genome-wide association studies, how the genes, that is traits, characters are associated with each other, right? Then candidate gene discovery. If you are looking for some particular, that is for sweetness or aroma or color or resistant to some diseases like that. So that particular gene discovery also can be done using this marker, DNA markers, right? Then genomic selection across validation. That is, we are going for exploration for, we can take the example of this rosewood. We went for exploration of this particular species in Tamil Nadu, Kerala, Karnataka, and Andaman, etc. And we gathered 173 superior exemplars and we assembled in a germ plasm bank. So this germ plasm has to be validated. Morphologically, we validated. Biochemically, validated. But whether it is this quality is hereditary, it is genetical or not. So, okay. So this is having a stable value or not. So for taking this species to the next level, we have to do the validation. So for that validation, we need the marker selection. So particular genomic selection from the bulk, from the 173 germplasm, selecting the one particular step, it is to take in for breeding purpose, genetic breeding or tree improvement program, and we have to validate that particular genome, genetic resources. For that one, this molecular markers are used, right? This is one of our experiment, and so the details, this marker assisted selection, and also we can use for to tag the particular trait, that is particular gene. So by our naked eye we cannot find this is good, this is bad, we cannot answer, but only through molecular marker only we can answer this trait can be heredited, this trait can be taken for next level, this can be tagged. So that for that we have to do this molecular marker studies, right? So the genetic distance between the species we can study. So it is very much important. So we no need to take the very closely related species. In that type, the variation is very narrow. So we have to choose the genetically distance uh, varieties for crossing or breeding or improvement program, right? So here it is written the rice varieties, 4 dozen, how they are related to each other. So this normally we use the simple sequence repeats, that is microsatellite markers, codominant marker for assessment of genetic variations in population studies, and to characterize, to get the passport data of a germplasm, and to identify the particular genotype, and for identifying a particular candidate genes also we use, and for species or individual identification, when morphological characterization fails, that time we have to go for this molecular classic characterization. Okay, so DNA from any part of an organism, we know we are all science students that can rapidly identify that particular species. So when morphological taxonomy is not giving a handshake to us, that time we can directly go to the molecular method. So there is a NCBA, and there millions of molecular data is available. So if we are taking the DNA and comparing with that, we definitely the sequence, if we are doing the sequencing, and when we are comparing the sequences with that, immediately we comes to know about that this belongs to which family, which genera, and it's falling nearer to which species, right? So immediately we can find out. Another method is the DNA barcoding method, that is derivation of short DNA sequences that is commercially available. That is there is a consortium for this one. So from them we can buy this plastics. So if Dasar is preparing a plant with particular character, he can tag this any of this plastic region. So if it is go to America or Africa, wherever it is, we can see when we are doing this molecular work, we immediately comes to know this is from the Dept of Botany college. So immediately we can come to know about that. So it's an universal fonts they are giving to us. They won't repeat the same barcode to any one of this group, right? So, like this, we did the characterization for the rosewood, that's the latifolia. We did the morphological variations for 173 CPTs, and we did the biochemical variations through yeah, TLC, HPLC, FTIR, GCMSMS, etc. And we explored all the components in it, and we found out the molecular variations so through different markers, right? So for that we have to do a lot of work. It is not an easy job. We have to standardize everything. Okay, for all the clones or individuals genetic resources, we have taken the particular same species, right? So once and for all 173 germplasms, we did this standardization of DNA extraction procedures, and quality and quantity, and the yield and the uneven temperatures and the primers, everything we have to standardize. Then only we will get the variations. So we did array-to-bit studies, we compared, we did inter-simple sequence studies, we did simple sequence bit, and we did SNP also. Then we could find what is the variations, which molecule, which addition deletion happened, and this is for which trait. So we done that up to that, and we deposited the transcriptomic data and whole genome data in NCBI. We deposited the metadata, and we did the genetic relationship distance and this genetic distance study also for all the 173 species what we have collected from Tamil Nadu, Kerala, Pondicherry and Karnataka region also. And we did the principal coordinate analysis, which population is having a very diverse populations, which is with more genetic traits. That is selecting a hero from a population, right? The hero's expression is excellent in all the ways. He physically looks good, and his expressions, actings, everything is good comparing us. So in that way, we are selecting the heroes from the population. So we are trying to see these variations. So among the regions, how much percentage variation is there among the populations, and among the regions, among the populations, among different trees, what is the correlation? Everything we are studying, and we are coming to the conclusion on the particular populations, and we are conserving that particular trees for further genetic improvement and breeding program to get a excellent germplasm for further work, that is to get a very good timber or to get a very good aromatic plant, or to get excellent antioxidant, excellent anti-cancerous activity compound rich plant. So likewise it goes on. So like that for Punkame peanut also, more than 91 germplasms we have assembled based on high oil content and high fruit yield, and we characterized based on morphological, biochemical and molecular base, and we assembled them. So we are taking the DNA, and we are running for its quality and quantity of DNA in agar cell. After this, what has happened? So you people might have think. So there is answer. There is a just documentation system in that. If we load the cell, it will copy all the bands, and it is having a special software. Through that we can store the 10 data, same as like the forward. Here also, if a band is present, it is 1. If it is absent, then we have to take it's a 0. So this scoring bands are very much on-zero data is very much important. This is the base for all the studies. If we take this one, and we have to apply this on-zero data for different software according to your utilization. Okay, so there are n number of these softwares are available in it. It can be Darwin, Harlequin, Bottleneck, structure. There are n number of softwares. So according to whether we are studying the diversity, population variations, or gene flow, or evolution studies, whatever it is. So we have to apply accordingly. Then we will get the our answers. So this is the characterization. So morphologically and biochemically and molecular wise, we have to do this work to get a halite. That is, in market we are getting a very good vegetables and very good product of our organic foods, right? So to get such a halite one, we have to toil, and we have to do the characterization work up to this much. Okay, and thank this characterization of Pongemia, as I told, we did the morphological, the part cells variation, the seed cells variation, oil content also variation, everything variation we recorded. And thanks to our team, and with this I conclude my lecture here, and I thank the GGDC College and Botany Department for their kind invitation. Thank you very much, sir.
Thank you,
Thank you,
madam.
madam.
Thank you so much.
Thank you for your
nice
nice
presentation.
presentation.
You
Thank you,
always
sir.
You
with
always
us.
with us.
Our next
Our next
presenter,
presenter,
our next
our
research
next
person,
research
sir,
person,
presenter Dr.
presenter Dr.
Anshuman Chando,
Anshuman Chando,
UGN PG
UGN
Department
PG
of
Department
Zoology,
of Zoology,
Raja NL
Raja
Khan Women's
NL Khan
College,
Women's
Mehnapur,
College,
West Bengal.
Mehnapur, West Bengal.
But I'm expecting.
Sir, are you here?
Sir, are you here?
Yes, I am
Title
hearing
of sir
Title of
clearly.
sir
presentation is
presentation
Aquatic
is Aquatic
Ecosystem,
Ecosystem,
Plants,
Plants, Animals and
Microbial
Microbial
Association for
Association for
Ecosystem
Ecosystem
Function.
Function.
Sir, what to
Sir,
you?
what to
Is it
you?
visible? Hello?
Visible.
Visible.
Hello, sir. Is it visible?
Visible.
Visible.
Yes.
মোবাইলে করতে জানে না বুঝলেন
Good evening. Good evening to everybody. Myself, Dr. Anshuman Chando, Associate Professor of Zoology, Raja NL Khan Women's College, Mednipur, Paschim Mednipur, West Bengal. First of all, I want to extend my heartfelt gratitude and thanks to the Lalgarh College, Government Degree General Lalgarh College, and also my
ওটাতে
friend
ওকে করতে হতো ওটাতে
Dr.
ওকে
Das
পাওয়ার পয়েন্টের সফটওয়্যারটা
and
ডাউনলোড করতে হতো দিয়ে
Dr.
তাকে
Ghosh
খেলালে ওটা
from
পড়তো
Seva Bharati
জানে না
College Botany
ধরে ধরে
Department
টানছে
for inviting me to give a lecture related to aquatic ecosystem, plant, animal, and microbial association for ecosystem function. As we know that a aquatic ecosystem is the 70%, near about 70% of the world ecosystem is aquatic, and the rest of the portion of the world is terrestrial land. So, aquatic ecosystem is very important and vital ecosystem where maximum amount of biomass are available in the world. And
হচ্ছে না বন্ধ হয়েছে
here, the function of the ecological system is very much complicated. There are so many types of aquatic ecosystem in the world. Basically, it is about freshwater ecosystem, marine ecosystem, estuarine ecosystem, and even within the freshwater ecosystem, it is about stagnant water ecosystem, or it is about running water ecosystem, lentic and lotic environment. That is the different uh, physical and biological aspects are there within the lentic and lentic and lotic environment aquatic ecosystem. Whatever it may be the ecosystem, aquatic ecosystem, there are some similarities between the ecosystem, aquatic ecosystem, like their importance within the aquatic environment, that is the biological diversity. Aquatic environment harbor most of them aquatic plants, aquatic animals, and microbial organisms. Both are integrated together to perform their ecological function to sustain the whole ecosystem. And as we know, the plants are the producers and animals are the consumers, as well as the microbial organisms are the decomposers. Not only microbial organisms, but also some other animals also acting as a decomposers, macro decomposers and micro decomposers. They are categorized into two categories, that is macro and micro decomposers. Macro decomposers are the benthic animals like crabs, mollusks, etc. They can crawl within the bottom of the aquatic ecosystem, and they can fragment the organic materials like leaf litter system of any plant products, and they make it fragments, small sizes of the organic materials, and then only the microbial organisms get a huge surface area, which is the place of their activity, which is the place of their action for the decomposition, and certain biochemical reactions within the aquatic environment takes place by the microbial organisms, both aerobic and anaerobic organism, microorganisms, fungi, starting from fungi to bacteria, all are actively participating for the recycling of materials, recycling of nutrients within the aquatic environment, from which the phytoplanktons get nutrients, and the majority of the organisms, animals, and fishes, amphibia, reptiles, mammals, etc., whatever it may be the animals present within the aquatic body. Within aquatic body, there are so many different type of animals, animal group of almost all type of phylums of animals group are available here. In some cases, some are adapted for the terrestrial or the muddy environment. They can rest within the flat mud flat also, and they are always take part within the uh, food chain of the aquatic ecosystem, and they serve as a part of their aquatic environment. Here we want to show a lake ecosystem, that is the terrestrial, that is the stagnant water ecosystem, where there are so many epiphytes, periphytes, and benthic plants are there within the environment, and as a whole, the natural uh, resource like sunlight, soil, water, all are there within the aquatic environment, and both biological components as well as the biological or the abiotic components are acting together to form a unique ecosystem here. Freshwater ecosystem, we are talking about the we will some make emphasis on the freshwater aquatic ecosystem, because majority of our population residing within the mainland. That is, I want to say that we are not majority of the population residing beyond the coastal line of the continent, and therefore we are facing some problems related to the freshwater environment. Majority of the freshwater body are is prone to the pollution effect. Basically, the anthropogenic pollutants are gradually editing the aquatic freshwater environment. Polluted pollution of the freshwater aquatic environment is created by the human being itself, and that's why we will give some emphasis on the uh, conservation of our freshwater aquatic body, as well as the wetland system of our terrestrial mainland body. And here the function of a ecosystem is done by the different group of organisms, where aquatic body is has a certain basin, soil basin, that will harbor some microorganisms, some benthic organisms, and the periphyton, epiphyton, and other like phytoplanktons and zooplanktons organisms are there within the aquatic environment. They are the major component of the aquatic ecosystem, and they are the primary producers, and fish, mollusks, aquatic mollusks and fishes are the major consumers, and some aquatic birds, amphibians, etc. are there within the aquatic ecosystem. They are the also consumers, secondary consumers, and altogether they are forming a compact, sustainable aquatic environment in the freshwater system. And now we want to characteristics of characteristics of an aquatic ecosystem. Factors such as temperature, sunlight, oxygen, nutrients determine which organisms.
কোথা থেকে বেরিয়ে গেল তো বেরিয়ে গেল নাকি? হ্যাঁ, তো বেরিয়ে গেছে নাকি? Dr. Chandra,
are you here?
maybe
Maybe network issue.
Please join.
Sir.
सेवा खुले किसी का मतलब नंबर होता ये हेलो कंसुमा join. Oh, मुश्किल कांड Dear participant, please wait. They're contacting. Sir is trying to
join.
হয় বলে কি মুশকিল কাণ্ড না নাইন 9326 75 019 75 019 Namaskar. 93820 93820 93820 80 934 80 934। अरे ढुको बेरी करे जी अरे ढुको बेरी करे जी কোথায় তুমি বেরিয়ে গেছো নাই তুমি নামে বলে বলে যাচ্ছো আবার বলো আজকে জানি করো তুমি আবার লিংকে জয়েন করো লিংকে জয়েন করে আবার স্লাইডে শেয়ার করে ওই জায়গায় চলে যাবে নাই ওই বলছি আমি তো একটু বলে না বুঝে আছে আরে কি হলো ব্যাটা দিয়ে
Hello,
হ্যাঁ তখন ঢুকছি
hello, hello. শোনা যাচ্ছে না?
Yes, sir, you are audible.
Hello,
You are audible, sir.
hello,
Hello. Yes, sir, you are audible.
hello. Is it visible?
No, not visible.
Hello,
No visible. No PPT is visible.
hello,
Just we are listening to you, but no PPT is visible.
hello, hello. Yeah.
Hello, sir. We are listening to you.
কি শোনা যাচ্ছে?
আমার একটু টাকা দিচ্ছে না বুঝতে পারছি না
Hello, hello.
Hello, sir. Your voice is audible, but PPT is not displaying.
Hello, hello,
Hello,
hello.
hello, sir. Please share your PPT.
Am I audible?
Yes, yes,
বলতে পারছি না
Hello,
yes, sir. You are
am I audible?
yes, yes. You are audible. Are you hear me?
Hello,
Hello. Are you hear me?
hello.
জনতা
বেরিয়ে গেছে কি করে? ঢুকেছি তো। Hello,
হ্যাঁ হ্যালো
হ্যালো
am I audible?
হ্যালো অঞ্জন স্যার
Hello,
হ্যাঁ start your
is it
presentation.
visible?
No, slides are not visible.
বোঝা যাচ্ছে না। Hello.
हाँ
Sharing screen. Share it. Hello. Share screen.
not
Please.
visible.
Shahi. Okay. Hello.
Not visible your screen. Maybe your network issue.
Hello. Hello.
Yeah, you're trying but not visible, sir.
Slide is not visible.
Now slide is not visible. Audio, where voice is audible.
Is it now visible?
No, no,
Whatever it may be.
no,
Can I finish?
not
Hello.
visible.
Am I audible?
Audible, but not your slide is visible.
So I want to conclude the talk. Uh, as it is not visible.
Okay.
Just I want to conclude that aquatic ecosystem is very important ecosystem, and as it is the location of the aquatic ecosystem from the landscape, that it is the lower level portion of the land morphological phase, and so the each and everything is dumping within the aquatic ecosystem, and the aquatic ecosystem is gradually deteriorating its quality. It should be measured biologically as well as the chemical methods. Biologically, it can be measured by the measuring the biological diversity, or the biological indexes can be used for the measurement of the water quality as well as the ecosystem quality. More the diversity, more the sustainability of the aquatic ecosystem is when the diversity is decreasing, the sustainability of ecosystem will be decreased very quickly, and it can be converted into another ecological system that may be that is known as the ecological suppression, and thus one aquatic ecosystem can convert it into a wetland as well as the mud flat, and ultimately it can convert it into a terrestrial land or the forest ecosystem area. So if we can think about the aquatic ecosystem of a coastal belt, or the estuarine ecosystem, where in the tropical country there are a forest relationship within the aquatic ecosystem, that is known as the mangrove ecosystem. Mangrove ecosystem is the very determinant factors within the estuarine ecosystem, where every day some part of the forest is inundated by the marine water body, as well as in a day there are two inundation within the mangrove ecosystem, and the leaf litter system is gradually drawn back to the estuary region, to the marine ecosystem through estuarine region. That's why the aquatic environment of the estuarine ecosystem is very rich with carbon, organic carbon, and it can it can very productive ecosystem. Very productive ecosystem of the world. Estuarine ecosystem is a very productive ecosystem, and the majority of the marine organisms as well as freshwater organisms come back to the ecosystem, and here they use estuary as a breeding ground or the nursery ground of that particular organisms, and they grow their juvenile organisms into a certain amount of larger sizes, and then after that they can back to the ocean, or as well as to the freshwater ecosystem. So estuary is the gateway. GT for migratory organisms. Migratory organisms, that means catadromous and anadromous migratory organisms can move passes through the estuarine ecosystem, and the estuarine ecosystem is the very productive, as well as the majority of the population who reside within the coastal belt. They utilize the estuary as their daily livelihood maintenance, and they can earn a certain amount of economic economic uh, mm, uh, mm, daily requirements they can maintain by the fishing or by using the estuarine ecosystem. So whatever it may be, the aquatic ecosystem is very important ecosystem. We have to protect it, maintain it, and sustain it throughout the year. Thank you everybody. Thank you for your patient without slide. Thank you, sir. Hello.
Thank you, sir.
Thank you, sir.
Thank you for
Thank
your
you for your
talk.
talk.
I think
I think
I think
I
this
think
this
this
presentation is the
presentation
last
is the last
presentation. So
presentation. So
we'll move to the
Mute হয়ে গেছে। আপনার মাইকটা মিউট হয়ে গেছে মধ্যবাবু।
Thank you,
thank you,
sir.
Amsuman sir.
This is the
This
third
is the third
presentation, and
presentation,
this is the
and this
last
is the last
presentation by the
presentation by the
resource
resource
person. So
person. So
we'll move to
we'll
the
move to the
next
next
presenter
presenter
session.
session.
Already I
Already I
mentioned there.
mentioned there.
Now this
Now
session
this
is for
session is
Dr.
for Dr.
Vinuta,
Vinuta,
Dr. Vinuta
Dr.
R.
Vinuta
Bhatto,
R. Bhatto,
head of the
head of the
departments of
departments
Zoology
of
and
Zoology and
Genetics,
Genetics,
Jyoti Nivas
Jyoti
College,
Nivas
Bangalore,
College,
West
Bangalore,
Bengal.
West Bengal.
Madam will chair
Madam
the
will chair
session
the session
to
to run the program.
and
And
in this
in this panel discussion,
many teachers
many
are
teachers are here.
Dr.
Dr.
Vidyanatha Jha
Vidyanatha Jha sir
from
from
Darbhanga,
Darbhanga,
Bihar,
Bihar.
Professor Dr.
Professor Dr.
Ravindranath
Ravindranath
Bhattacharya from
Bhattacharya from
Bardhaman.
Bardhaman. Sir is
Retired
retired professor of life science,
Presidency
Presidency University,
Kolkata.
Kolkata.
Dr.
Dr.
Majita
Majita
Begum
Begum
from
from
Theni,
Theni,
Tamil Nadu.
Tamil Nadu.
Dr.
Dr.
Maheshwari
Maheshwari
from
from
Zoology
Zoology
Department of
Departments of
Bhubaneswar
Bhubaneswar
Singham
Singham
University,
University,
Tamil
Tamil
Nadu.
Nadu.
Dr. V.
Dr. V.
Gayatri,
Gayatri,
Department of
Department of
Botany
Botany of the same
Tamil
University,
Nadu.
Tamil Nadu.
Dr. R. K.
Dr. R. K.
Misra
Misra
from
from
Biodiversity
Biodiversity
Conservation
Conservation
Department,
Department,
Maharaja
Maharaja
Bhanjdev
Bhansdev
University,
University,
Takatpur,
Takatpur,
Odisha.
Odisha.
Dr. P.
Dr. P.
Ghosh
Ghosh
from Seva
from Seva
Bharati
Bharati
Mahavidyalaya,
Mahavidyalaya,
Jhargram.
Jhargram.
Santosh
Santosh
Murmu sir
Murmu
from
sir from
Raja N. L.
Raja N. L.
Khan
Khan
College.
Women's College.
Dr.
Dr.
Virasar
Virasar
Bera,
Bera,
Department of
Department
Zoology
of
from
Zoology
St.
from
Joseph
St.
College,
Joseph College,
Darjeeling.
Darjeeling.
Dr.
Dr.
Sumitra
Sumitra
Salam,
Salam,
Assistant
Assistant
Professor,
Professor, Departments of
Botany,
Botany,
Nambol,
Nambol,
L.
L.
Sanoi
Sanoi
College,
College,
Manipur.
Manipur.
Dr. K.
Dr. K.
Maji,
Maji,
Head
Head
Department of
Department of
Zoology,
Zoology,
GGDC
GGDC
Lalgarh.
Lalgarh.
Dr.
Dr.
Sujit
Sujit
Isku,
Isku,
Assistant
Assistant
Professor of
Professor of
Zoology,
Zoology,
GGDC
GGDC,
Gopiwallapur,
Gopiwallapur,
Beliabada,
Beliabada,
Jhargram,
Jhargram,
and many
and
more.
many more.
So,
So
even
even
today,
today, Dr.
Vaishali
Vaishali
Bamsod
Bamsod
from
from
Maharashtra,
Maharashtra,
Pune
Pune
is present
is
here.
present here.
So,
So
I think this
I
is
think
a
this is
nice
a
session
nice session
to be
to be
organized
organized
by
by
our
our
Professor
Professor
Vinuta
Vinuta
Bhatto.
Bhatto.
Over to you,
Over to
madam.
you, madam.
Thank you, sir. So, sir, with your permission and with the esteemed panel, I would like to begin today's technical session of day 2 of this conference. Today, we have 15 presenters who will discuss their research work with the esteemed panel members. Now, before I begin, I would like to brief our participants about this session. I'll call out the names of the participants. You will be given 4 minutes time to brief about your research work, mainly focusing on the objectives and the aim that you had to achieve and the key findings. You can also emphasize on the future scope
What future directions do you envisage for this research, and how do you see it impacting the field? You can brief about any current gaps or challenges in the field and also potential benefits of your research. So, before any ado, I'll call up the first presenter. So, the first presenter for today is Konkana De, and her topic is benefit of medicinal plants in the treatment of different diseases. So, Ms. Konkana De, are you there? Ms. Konkana? I think she's yet to join. We will put her
Yes,
the.
ma'am, I am present here.
Yeah. Okay, you can start. Madam, you'll be given 4 minutes time and 1 minute for the discussion. So you can brief about your work.
Yes, good evening. Thank you, everyone. Thank you, the committee, conference committee, and all esteemed guests. I'm Konkona De. I'm presenting my paper on benefit of medicinal plants in treatment of different diseases. And am I my presentation is visible to you all?
Yes, it is visible.
Thank you. And in earlier conversation on topic, we came to know that plants are important part of environment. They have different physiological process, and because of the different physiological process, they produce food and which deliver to the every living thing, and they produce some chemicals which are very essential, and which biochemical are usually used in treatment of different diseases, and this study is done in
Madam,
yes sir.
do not actually show your slide. We do not actually want to see your slides. Just you present your different aspects and open your video so that we can take the snaps. Okay.
Yes.
Don't share your slide. That is not required. As per the question from the panel reviewers, they will ask the question. Just give your answer, appropriate answer.
Sir, can you help me? How can I open my video? I stop presenting, then I open my video.
You open your video. You open your video, but not present your slides. Okay.
So and now I
Camera.
stop
Open your camera. That is
presenting.
better. Open your camera to take the snaps. Okay. That you are talking something regarding this.
Yes.
No need for presentation.
Now I am visible.
You can turn your camera. Yeah, now you are visible.
And there are different medicinal plants which are Allium sativum. Generally, in Bengali we call it rosun. The bulk part of this plant are used for different antivenom uses. Bulk is made by the medicine is formed by taste and given orally, and there is mimosa pudica from Fabaceae family, leaves are ground and made into paste to infected areas. And there are different plants which and most of the plant whole body is medicinally used, like Azadirachta indica, leaves, fruits and roots, all are very useful for human beings. Turmeric, which is Cumula longa, generally called as Mulut in Bengali, which is very useful to in first aid, like cut, wounds, and in India, 80% of poor people and elderly people use this kind of conventional medicinal treatment, and we in this criteria, I want to say that the people need to conserve those type of plants and take care of do not indiscriminately use this plant that if we indiscriminately use those plants, they can become extinct. So should be careful about their usage and for their growth we need to plant those plants. And in this now it is winter season, people generally they suffering from cold flu and for this Atatara Basica in generally we call it Bashokpata in Bengali. Those leaves are very useful to treatment in cough and there are some weeds also which are very toxic effect. We should careful about these plants that those plants are not spreads around that cause asthma and different kind of disease. So we should take care of the medicinal plant and should give, uh, spread our knowledge about the uses of those plants and their conservation. It's all about my paper. Thank you for listening to me.
Thank you, Konkana. Now, can I ask the esteemed panel members to ask some questions or throw some insights?
Yeah. Konkana, I just wanted to ask you that the plant that you have taken for your research, was it not known as a medicinal plant before, or you have done some reference and then you are saying that it is a medicinal plant?
It is reference and some like turmeric and neem.
No, which plant you have taken for your research?
It looks like a review presentation.
Is it review or secondary data? Some plants are available in our locality which I am coming from. So I started on from.
Okay, so that this particular plant is already being used, but ethnically it is used as a medicine, no? Okay, ठीक है.
Any other questions? Shall we move on to the next presenter?
Yes, ma'am.
The next presenter is Soma Parshaha and Anisha Haldar, and their topic is solubilization of immobilized soil phosphates by hydrophilic Sundarban mud bacteria. So who's presenting, Soma Paul or Anisha? Okay, Soma Paul, you can start.
I am presenting you. Am I audible?
Yes.
Okay.
You can turn on your camera.
Yes, ma'am.
Yes.
My topic is solubilization of immobilized soil phosphates by halophilic Sundarban mud bacteria. And as we all of us are very aware about the plant health, when the agricultural field is a very extremely reactive phosphate when it is present, then always we will get the immobilized condition of that phosphate. Like when it would be precipitated with calcium ion or the magnesium ion or the ferrous ion, aluminum ion, and thereby the phosphates of these ions and become salts and it becomes totally immobilized. And that condition of the phosphorus is totally insoluble. Now, there are some microbes which are typically known as phosphate solubilizing microorganisms. They are able to solubilize those kind of salts. present in soil, and especially they can mobilize those salts via different programs, microbial programs. So that means the mechanism like chelation or the acid release. That means when the pH value of the soil becomes very acidic or lower, then thereafter the mineralization. But in our program, it would be only the enzymatic, that is the total biological system. And our target, that means the objectives, is to isolate some halotolerant phosphate solubilizing microorganism, especially the bacteria, from Sundarban post-monsoon soil. And you may ask why post-monsoon? Because the salinity during that period, as we have got the result, that was very high. And thereafter, to determine the salinity, effect of salinity on the phosphate solubilizing index. That means how much efficiency they have to solubilize the insoluble phosphate salts. And finally, we have to select one potent strain on the basis of this PSI, phosphate solubilizing index. Later on, we have already what we did, uh, determination of the capability of potent strain for the solubilization of different, four different types of phosphate salt, like the calcium phosphate, aluminum phosphate, iron, and the magnesium optimization are also done, like the carbon substrate concentration or the phosphate concentration for the synthesis of phosphatase enzyme, especially by the potent bacterial strain. And finally, our target was also what kind of enzyme it is, and for this, the temperature optima and pH optima, especially, we have determined. And during that period, the 11 very good strain we have taken out, and among them, one very good strain showed that this is very special because up to 6% of salinity it
Soma,
withstand, and
Soma, directly go to results and discussion because
yes,
you have
I'm
already,
going for that.
yeah, yeah.
F24 PM 101 from the Frizarganj area. That was PSI, that means phosphate solubilizing index was extremely high, 2.8, compared to others, other 11, 10 strain. And that was recorded as a very good non-sporulating long bacilli type organism, gram-positive strain. And thereafter, when we tried to justify the program of different parameters, then we have seen that the cellulose was a very good carbohydrate for this. So the
Yes,
cellulase present in the soil via from microbial action, and when cellulose is present, then it is showing the maximum amount of PSI. So from that viewpoint, it was nice, and thereafter we have seen this strain. F24 PM 101, it can able actually the all four aluminum phosphate, iron phosphate, magnesium phosphate equally. And with respect to their growth also, it is very nice. So we have tried to justify the concentration of phosphate also, and we have seen up to 0.2% level of phosphate. It was nice for all, and up to 0.5% of the calcium phosphate, it was excellent. So you can also ask that Pickup media, what purposely we have used, that basically contains 0.5% of calcium phosphate. So it is very comparable with that. And thereafter, we have tried to justify the optima, pH optima and temperature optima. And interesting thing is here, it is the acid phosphatase, and the pH 4 was very good for that. Okay, and temperature, it is totally mesophilic. 30 to 40 degrees centigrade was nice, and the maximum was find out as 35 degrees centigrade. So in conclusion, among the 11 selected phosphate solubilizing bacteria, most of them are tolerant up to 2% sodium chloride, but more than 50% strains are also showing the tolerance level up to 6% of salt in their growth medium. And our most potent organism, F24PM101, had been selected on the basis of its PSI, and 4 tested inorganic phosphate salt. And later on, we have also used some organic phosphate, organophosphate. So that is also okay for this organism. And phosphatase synthesis was good up to the level of 160 unit, and sometimes it is much more on the basis of its carbohydrate concentration when it is increased higher. And
Soma, Soma,
yes, please.
Soma, please stick to the timings. You have 1 minute. You have exceeded the time, but still 1 minute to complete your presentation.
Okay, okay. I have just now completed.
Yeah.
The hydrophilic bacteria, this kind of organism really would be very promising biofertilizer, especially for the saline soil like our Sundarban agricultural field. And I must acknowledge DBT for our financial support, and some references with me, but it is not showing here. Thank you, ma'am.
Yeah. Any questions?
আস্তে আস্তে শুরু করে দিই ডাউন আস্তে
Soma,
Yes, please.
how can you carry this research results to field actually? Have you
Actually,
looked into benefit?
we have visited 5 to 6 spots, and five spots result we have actually presented here. And these are the different spots like Canning, Dobaki, Frazerganj, Gachkhali, and Kumirmari area. And via big ship, we have actually three different departments project: microbiology, zoology, and botany. And all of us are going to visit for collection of the samples.
Panel
Hello,
members?
Soma.
Yes, please.
Hello, Soma. I just wanted to know the duration of the study.
Uh, it is the post-monsoon sample of 3 years.
3 years. Why can't you try to outer sides of Sundarbans forest? Because most of the seashore areas has that salinity region, no? So you can
Madam,
use
please
that
be louder.
strain. Hello?
You have
Am I audible?
Yes, please.
I think you are restricted to that Sundarbans only. You may go to other seashore also. This is my suggestion.
Okay, ma'am.
I think this is nice work. Then you may go for other areas also because that tolerance should be induced for the surplus vegetation near the seashores. That's why uh, I wish to just
Okay,
justify to
ma'am.
you. Thank you.
Sure. Thank you, ma'am, for your suggestion.
We will move on to the next presenter. Next presenter is Karan Saran, and the topic is environmental assets in and around Kanyadoba of Jhargram with special reference to ethnic community based knowledge.
Madam,
Yes, sir.
Karan
Karan
Soren
Soren
is
is
my
my
student,
student,
but
but
he is
he is
from
from
humanities
humanities
department.
department.
Okay,
Okay,
Okay.
but
but
many
many
years,
years,
2 years,
2 years,
he's
he's
attached with
attached with
such kind
such
of
kind of
environmental
environmental
research.
research.
And
And
Konyadoba is
Konyadoba is
a
a
small
small
village,
village, tribal
community-based
community-based
village
village
of
of
Jhargram
Jhargram
district,
district,
nearer to
nearer to
Jhargram,
Jhargram,
near
near
about 10
about 10
kilometers
kilometers
apart from
apart
the
from the
Jhargram
Jhargram
town.
town.
So
So
he
he
recorded
recorded
many
many
assets
assets
of
of
his
his
own
own
village.
village.
Say for
Say, for
example,
example,
many
many
medicinal
medicinal
plants,
plants,
many economically
economically
important
important
plants, many
plants, many
agricultural
agricultural
crops,
crops,
and so
and so
on.
on.
Even
Even
animal
like
butterflies,
butterflies,
birds,
birds,
insects, and
insects,
so
and
on.
so on.
Some
Some
mammals.
mammals.
So
So
physically he
physically he
presented
presented
paper
paper
on
on
20th
20th
in
in
college. Okay.
college. Okay.
So this
So
is
this is
physically
physically
presented. The
presented.
paper
The
is
paper is
physically
Okay.
physically
presented.
presented.
Okay. Shall we move on to the next presentation, sir?
Yeah.
Yes.
Yeah.
Yeah. Next presentation is by NG Mahajan and Chopra. So their topic is in vitro antimicrobial activity of herbal composite. So can we have the presenters?
Yes, madam.
Yeah.
Okay. Good evening to all of you. Uh, Today I am presenting the research work entitled In vitro antimicrobial activity of the herbal composite. Madam, on the basis of traditional use and ethnobotanical knowledge, for uh, for the present study, we mainly select the plants for the polyherbal mixture that have the nephroprotective effect, because there are no antibiotics that are inherently nephroprotective in sense of both the treating infection and preventing the kidney damage. The selected six herbal plants, that is the Buhnia resmosa. Uh, we use the stem bark of this Buhnia resmosa, then Dolichos biflorus, seeds of this plant, then flowers of Spiranthes indicus, seeds of Tectona grandis, and leaves of Teprosia purpurea, fruits of Tribulus terrestris. And the tested organism, for that, 5 gram negative bacteria, that is the Pseudomonas aeruginosa, Escherichia coli, Proteus vulgaris, Proteus mirabilis, and Klebsiella pneumoniae. With this 2 gram positive bacteria, that is the Staphylococcus aureus and the Bacillus subtilis, 2 fungal strains, that is the Candida albicans and the Aspergillus niger. Then, after carrying the in vitro antimicrobial assay by the agar-Bell diffusion method, we got the significant result against the control. Uh, our result mainly show the highest inhibition zone was measured against the Proteus vulgaris, and least inhibition zone was observed in against the Klebsiella pneumoniae at the 1000 microgram per ml of the polyherbal mixture. For this antimicrobial assay, we use the standard dose, that is the gentamicin, at the concentration of 10 microgram per ml. However, the both fungus strain, that is the Aspergillus niger and the Candida albicans, at the 1000 microgram per ml of the polyherbal mixture show the same millimeter zone of inhibition. And on the basis of our result, it may concluded that the herbal mixture possesses a significant antimicrobial activity and may be of the use for the development of the phytomedicine for the therapy of the infectious diseases. Thank you.
Any questions?
Yes, ma'am.
Yeah.
Number, actually, I want to know that are you using stem bark as a sample, right?
Yeah.
Yeah. Actually, there will be a chance of presence of pollution in this sample. How did you overcome this issue? I think
Uh,
purification is the problem here. How did you overcome?
After thoroughly washing, madam, thoroughly wash that stem bark under the tap water.
Right. But still, I think here the purification is the problem because stem bark is mostly affected by the pollution frequently, and it will affect the functions of stem also.
Okay.
So I think go for that pollution study also.
Okay, definitely.
Thank you. Thank you.
Vinita ma'am,
Vinita ma'am,
Yes. We'll move
Dr.
on to.
Madam,
madam,
Yes, sir.
Dr.
Dr.
Rajani
Rajani
Gautam is
Gautam
not
is not
feeling
feeling
well,
well,
so
so
now he
now
will
he will
deliver
deliver
the vote
the
of
vote
thanks
of
for
thanks for
resource
resource
person
person
session.
session.
Okay,
Just
sir.
Just
allow
allow
her
her
We'll
to
hold
do
on.
this.
Yes. Okay. Okay.
Now Dr.
Now Dr.
Rajani
Rajani
Gautam will
Gautam will
deliver
deliver
vote of
vote
thanks
of
for
thanks for
resource
resource
person
person
session.
session.
Okay.
Okay.
Okay, sir. Very good evening to all dignitaries present here. I, Dr. Rajni Gautam, Professor and Head, Sam College, Bhopal, pleased, privileged to be the part of this August gathering. My sincere thanks to Dr. Debrata Das for imparting me this opportunity to deliver vote of thanks this evening. It's really a pleasure that Dr. Debrata and Dr. Pampi Ghosh organize such a useful and knowledge-packed webinars every year on a new and enlightened topics. These webinars not only provide the participants in-depth content but also are full of insights. Once again, I congratulate organizers for their sincere and excellent efforts. And last but not the least, I would like to thank all the resource persons who, in spite of their busy schedule throughout the day, have taken time and attend the program and helped to make this conference a great success. It was really difficult to organize this conference without their help and cooperation. Thank you again. Thank you, Dr. Devrata.
Thank you,
Thank you,
madam.
madam.
Thank you.
Thank you.
Thank you, ma'am. We will continue the presentations. The next presentation is determination of antimicrobial property of Panax pseudogencin from adjoining areas of Darjeeling Hills. The presenters are Sujata Kalikotai and Bind Chandra Sharma. Do we have the presenters?
Hello.
Yeah, who's presenting? Sujata. Okay, you can
Okay.
start.
Good evening. Okay. Good evening, one and all here. Uh,
Yeah, you are audible. You can start.
okay. Okay, ma'am.
Could you please turn on the camera?
Okay, ma'am. Is it essential to turn on this camera,
If
ma'am?
it's comfortable, or else you can carry on.
Okay. Okay, ma'am. Okay. Since I am out of station, ma'am. So
Yeah, please
can
proceed.
I start, ma'am?
Yes, yes.
Okay, ma'am. Okay. Okay, ma'am, the topic of my presentation today is determination of this antimicrobial property of Panax pseudoginseng from adjoining areas of Darjeeling Hills. Uh, to start, the antimicrobial, the objective of my study is to determine the antimicrobial activity of Panax from Darjeeling Hills. Mm, okay, so historically, a large portion of the world's medicine it has been derived from plants. Mm. So, my main study aims in main study aims in determining the uh, antimicrobial property of Panax pseudoginseng of the Himalayan region, mainly of the Darjeeling Hills, and as limited information is available about the plant specimen from Darjeeling
ক্যামেরাটা
Hills,
খুললে সেটা
and
বলতে
that
পারছি
is
না
Panax pseudoginseng.
ক্যামেরাটা না খুললে হয় এটা যে
The
প্রেজেন্টেশন করছে
area
রেকর্ড
of
হচ্ছে হচ্ছে না
collection of this plant species, it is from Ragivin area, which is about 20 kilometers away from main Darjeeling town. The materials and method uh, which has been adopted in determining this antimicrobial activity I have mentioned in my slides. The test microorganism which has been taken is I've taken 8 microorganisms that includes both gram-positive and negative bacteria and a fungus that is Candida albicans. So the screening of the antimicrobial activity, it has been performed following the method of Bauer et al. And in this figure, you can see the image of Panaxidoglycin, which has been taken. The extract of this plant, it has both antiviral and antibacterial activity, as it can be seen in the image. So from the table which I have presented in with this PowerPoint, you can see that the panaxidoglycin, it is the ethanol and methanol extract of panaxidoglycin was taken, and it is seen that the ethanol the sterile distilled water has been taken as a negative control in this case, and streptomycin is taken as a positive control. We could see that both the ethanol and methanol extract of panaxidoglycin was active against gram-positive bacteria and gram-negative bacteria and the fungus as well. So from this, from our work, it can be concluded that the results which were obtained from our studies now it may be helpful in developing the plant-based natural antimicrobial agents like fungicides for preventing and curing the common diseases of humans and to reduce the pathogen population. But further research and clinical trials are essential. So, thank you, ma'am.
Thank you, Sujata.
Yes, ma'am.
Yes, no. Any questions?
Hello, Sujata. Hello.
Yes, ma'am.
I just wanted to.
Hello. Yes, ma'am.
Can you hear me?
Hello.
Hello.
Yes, ma'am. I can hear you.
Yeah, just.
Yes, ma'am. I can hear.
Okay, I just wanted to ask you one question, one simple questions. Has this panaxidoglycin been traditionally used by local communities in the Darjeeling Hills for healing infections? Do you have any idea about this?
Yes, ma'am.
Or is there any?
Yes, ma'am. I have.
Okay, okay. Is there any conservation concerns for this species? Due to over harvesting or just any policies there to protect?
Okay, ma'am. Okay, thank you for the question, ma'am. Ma'am, in this Ramlevan area, there are some small nurseries which has been made by private people. For in the personal nurseries, they have preserved some of the panic species in the personal nurseries, especially in this Ramlevan area, ma'am.
Okay. No, sir. Thank you.
Thank you, ma'am.
Thank you. We'll move to the next presentation. The title of the presentation is A Brief Discussion on Boswellia serrata, an important medicinal plant. And the presenter is Soma Chanda. Ms. Soma, are you there? Soma Chanda?
No.
Soma? I think Soma is not present. we'll move to the next presentation, that is by Anvesha Ghosh and Debabrata sir. So the title of the topic is, yes sir.
Madam, physically
Madam, physically
presented at
presented
college.
at college.
Okay,
On 20,
okay.
20.
Sir,
20.
this Soma is not there.
Maybe
So,
absent,
Maybe
maybe
absent.
absent.
Maybe absent.
okay. So we'll move to the next one, that is in vitro propagation of Docus carota, a key to mass propagation. And the presenter is Deepu Samanta.
Yes, ma'am.
Yeah,
Good evening to all.
you can
Uh,
start.
Okay, so thank you. Uh, Myself, Dr. Deepu Samanta. My title is the topic of my today's presentation is in vitro propagation of Daucus carota, a key to mass propagation. As we all know, that in vitro propagation has become an essential tool in modern biotechnology. So in vitro propagation can be an alternative method for propagation. So here my objective is the clonal propagation using different tissue culture techniques in Daucus carota. Here I have followed the methodologies in vitro seed germination, shoot bud multiplication, micropropagation, callus-mediated organogenesis, and somatic embryogenesis. Here we have followed 5 stages of micropropagation, that is starting from stage 0, stage that is selection of mother plant and maintenance, stage 1 initiation and establishment of culture, stage 2 shoot bud multiplication, stage 3 root induction, and stage 4 that is acclimatization. Here I have the video PPT where I already sent. Here I have already mentioned the table which shows that after 28 days 90% of radicular growth and 100% of radicular growth and 90% of premular growth was obtained in case of in vitro seed germination. When we compared the rate of shoot bud multiplication using BAP hormone, it is seen that BAP 2 mg per liter hormone was the best for shoot bud multiplication. When we go for the callus mediated organogenesis, so for there are 2 phases, that is callus induction and another one is callus to regeneration, and that is shoot and root regeneration. For callus initiation, we have used 240 1 mg per liter hormone, and after subsequent subculture, when callus was subcultured in MS media without any hormone, then shoot and root formation was there. Callus mediated organogenesis is also known as indirect organogenesis. Then come forward the somatic embryogenesis. Here we have selected the explant as internode. Then callus formation was in the 240 1 mg per liter media. Then when withdrawal of 240 hormone, it shows that initiation of different stages of somatic embryo development, and then somatic embryo maturation. That is, there are certain stages of somatic embryo development like globular stage, followed by heart shape stage, torpedo stage, bipolar stage, and finally the last stage is in plant-like formation. So these are the 3 techniques only. Here I have shown that callus mediated organogenesis, somatic embryogenesis, and micropropagation. So it can be concluded that the protocol we have used is very effective and easier for rapid production of in vitro regeneration of Daucus carota.
So, thank you. Hello?
Yes, ma'am.
Yeah. So, she has concluded. Can we have any questions?
Presentation হয়ে মুগ্ধ হচ্ছে বলে
Panel members?
No, ma'am.
Shall I proceed? Okay. The next presenter is Kirtana. There are two actually. The presenters are Kirtana and G. Maheshwari. The topic is formulation of herbal toothpaste and evaluation of its antimicrobial activity.
Yes, ma'am, I'm present, ma'am.
Yes, you can.
Yes, ma'am. Thank you, ma'am.
Kirtana.
Pleasant evening to all. Myself, Kirtana, research scholar from the Department of Zoology, Agnashingam Institute for Home Science and Animal Education for Women, Coimbatore. So, I'm doing my PhD under the guidance of Dr. G. Maheshwari, Assistant Professor, Department of Zoology. So, actually, my title is on formulation of herbal toothpaste and evaluation of its antibacterial activity. So, basically, introduction about the herbal toothpaste. Usually, we use the commercial products what we get in our market. So these they are available in the form of paste and gel, etc. for keeping our oral health in proper. Uh, so our oral health is more important for us because the oral health what we having we are taking in our it will enter into our stomach and it may cause many problems. Many microorganisms are entering only in the form of by when we are eating or when we are speaking all this time which is present in air will enter into our stomach. So we should keep our oral health in a proper way. So when we go for a commercial product, the commercial products are having more high toxic chemicals, which is used to help us to keep our oral health. Of course, we have some herbal toothpaste, but they are using some preservatives to preserve. That also has a chemical things may cause some problems in later times. So keeping all this in facts, I have done a formulation, a herbal toothpaste formulation using a lemongrass extract, which lemongrass has a high antibacterial activity and antimicrobial activity against some microorganism, even a bacteria and fungus. So, and other some plant materials I also used in my formulation because this plant materials what I choose for doing is like turmeric and cardamom, coconut oil, essential some essential oil and cinnamon, all these things because these are having an anti-inflammatory, antibacterial thing which is commonly available in our kitchen. So, which keeping all this, I have done a toothpaste formulation and I have done the antibacterial activity of this lemongrass extract. I made a lemongrass extract using acetone solvent. So by this, the particular bacteria is Streptococcus pyogenes. This bacteria is been causing a throat infection which enter through our oral form. So this particular bacteria is cause many health issues to our body. So this lemongrass has an antibacterial activity against this Streptococcus pyogenes. So and when I'm going to my result, this formulation, I compared my herbal toothpaste with the commercial available toothpaste. I'm not exactly mentioning that commercial toothpaste, but I took two, which is usually we are using in our home. So when I compare to that, the formability of this our toothpaste what we made in the laboratory doesn't have because we didn't use any surfactant or any chemical materials for it. But what we get in the commercial products are having more formability. That means that may cause some ill gum problems and teeth issue, the enamel decline. All these problems may come at a later age, but now we don't know. But later age it may cause. Even some people may swallow their toothpaste. It is because of that also we can get problem. But this herbal toothpaste, if we swallow also, it not happen because these are some having medicinal property and may not cause that much big problem to our health issue. And the antibacterial activity also showed highest. Yeah, I have taken three different concentrations. In the highest concentration, each concentration also showed an zone of incubation against this Streptococcus pyogenes. So by this overall work, I am concluding that though we have many commercial products, we can prepare our own herbal toothpaste in our homes, which we having in the kitchens and all these things. We can't keep it for a long day, the shelf life, but we can prepare according to our days and we can use it. That we can keep our oral health properly and even eco-friendly, and it may not affect our ecosystem. And these commercial products also will not affect you. The chemicals also will be. We can prevent us from this, and we can take care of it. So thank you, ma'am, and thank you all for giving me this opportunity. And a special thank to the organizing committee for giving me this opportunity to present my work. Thank you, ma'am.
Shall we take some questions?
Kirtana,
Yes, ma'am.
uh, um, in your presentation, you said about that you can prepare it at home. Did you use it? Did you use your own preparation?
Yes, ma'am. Yes, ma'am. Actually, we prepared, and among us, students, and all we used it. It gives a good result comparing to our commercial product. We get some. When we use our commercial product, we get some. Even some people get pain or some type of burningness. But when we use this products, we don't get that kind of any problems because this cardamom gives you good breath smell also. We cannot get
Mm.
any bad smell with this
Okay.
all.
How long you have been using this?
Ma'am, this work I have done recently from this 1 year because
Okay.
my field is different from my work is differ. But when as I'm a research scholar, I am doing my work is on stone drain test. But usually we used to do our daily work. When I we use social media, I just thought, why can't we prepare a whole herbal toothpaste? So on this, I studied some review paper and I done this work as a small work. So I'm continuing using it, and we were
Okay.
students
Okay.
who together with yeah.
Have you labeled your product?
Ma'am, not yet, ma'am. Just I done this work. I should go
Okay.
with my authorities. I have to get some permission. So
Acha, acha.
all this I have
Acha,
to process.
in your presentation, you have also said that it is a herbal toothpaste, but without any preservatives
Yes,
added in
ma'am.
it.
Yes, ma'am. Yes, ma'am.
So
Yes,
how
ma'am.
long that will last if you prepare
That's,
today?
ma'am, that's what, ma'am, this shelf life.
Hmm.
When I prepare, ma'am, I use some coconut oil. I didn't use more distilled waters. So if you use
Acha,
more waters,
right.
the organism may come and fungus
Yeah,
infection,
yeah,
all this
yeah.
stuff. This type of oil, it may preserve for a long time. So that's my,
Okay.
yeah, I thought, what let me do with the oil and give
Acha.
some preservative.
Okay, okay. Continue with your work. Good.
Oh, thank you so much. Thank you, ma'am. Thank you, ma'am. Yes,
Kirtana,
ma'am. Yes,
uh,
ma'am.
is there any possibility of this extract being used as mouth freshener or something, or composition to use as, because all these days whatever the mouth fresheners that we are getting, it is having a lot of side effects. So could it
Yes,
be?
ma'am. Yes, ma'am. Ma'am, actually, the lemongrass has been used. There is some work has been done using as a mouthwash, the lemongrass oil. So usually we can do oil pulling using ginger oil and coconut oil. These are good mouthwash. I refer it because usually from our small age we use this coconut oil and ginger oil. Likewise, we can use this lemongrass extract. But going with a solvent extract, we can prepare as an oil and we can use it, ma'am. That is good for as a mouthwash comparing to our commercial products.
Yeah, good. Very good presentation.
Okay. Thank you so much, ma'am. Thank you.
We'll move to the next presentation. The title of the presentation is The Role of Farmer-Led Selection in the Evolution of Traditional Rice Varieties. So the presenters are Meetu Day and Santi Ranjan Day.
Yes.
Yes, ma'am.
Okay. So, respected chairperson, dignitaries, and other participants, a very good evening to all of you. The title of my talk is The Role of Farmer-Led Selection in the Evolution of Traditional Rice Varieties. The reason I chose that was during my doctorate days in Bose Institute, I worked with nearly 180 traditional rice varieties of West Bengal, only of West Bengal, and also other like rice varieties from the other countries, which were procured from the International Rice Research Institute. But after working for a long time in this project, there was one question that came to me after all the thesis was done and all that. And that was what leads to selection. Because in my course of studies, I had found that there were some varieties which did not yield much. They don't have any very good characters also in terms of fragrance or aroma or yield or tillering ability, but still the farmers kept it. So, so much variety is there, but all of them are not yield related. And since I was working only with land races, it means traditional rice varieties that is naturally evolved. But the hypothesis was that there was some external criteria also, which was it must be a farmer-led selection. So the traditional rice was there. It was a land race, but the external factor was farmers' selection criteria. It could be cuisine, it could be tillering ability, it could be deep water submergence tolerance, or it could be salinity tolerance. So something led to the farmers to keep these varieties present in the normal gene pool. So these are still present in West Bengal, but in very small pockets and in marginalized lands. So what we did was we took 10 characters, 10 important characters, and we had took 30 rice lines, this for the study. They were some commercially aromatic rice like the Baksha Bhog, Govinda Bhog, which is commercially available. And we took some non-aromatic rice varieties also. And so some of the varieties were natural land races, some of them were commercial varieties, and some of them were somewhere in between. And then we applied and we did the agromorphological characterization, and we took 10 crucial agromorphological traits, of which yield was very important. The other one was ecological selection. And then we put it into a multivariate analysis using Plastris, which is a web tool. And then what we got a heat map, which showed that the farmers' selection criteria, like those which were not only related to yield or other factors, those also were important in the selection. And that led us to the conclusion that farmer selection, that is farmer selection during artificial selection, has a role in the evolution of the varieties which we call as traditional land races. So it's not what we are trying to say. It's nature is of course there, which led to the evolution of these. But because rice is there for thousands of years, so the input of the farmers' selection criteria also matters. So we will in future. So this was only 30 rice lines. I have the data for 180 rice varieties, traditional rice varieties. So I'll be using all of them maybe later on to get the heat map, and then maybe we'll come to some conclusion. I don't know what, but let's see. So the future seems exciting. So thank you everybody for your patient hearing, and I'll sure share my findings later on. And Dr. Devabrata Das and Dr. Pompi Ghosh is here. So through them you'll get to know of the new findings too. Thank you so much.
Thank you, ma'am. Can we have some questions?
কেন রেডি আবার নতুন করে শুরু করতে পারতে না এটা ছোট ছোট করবি এবারে বলে ভালো
We'll move on to the next. Yeah, we'll move on to the next presentation. Next presentation is the composition of gust microflora of stingless bee, Tetragonella, from Darangao Taluk of Jalgaon district. The presenters are Varun, Hemant, Namrata, and I don't know who's presenting. May I know who's presenting?
Yes, madam. I am Dr. Manoj Kumar Chopra presenting the presentation.
Yes, sir. You need not share the PPT. You can orally discuss.
Okay, thank you. So my topic is, madam, before going to start the presentation, I welcome to all the dignitaries and the participants. Very good evening to all of you. And I am thanks to the Devarabha Das sir for giving me such opportunity to present presentation in the conference. The topic of my presentation is the composition of gut microflora of stingless honeybee, that is Tetragonella species from Dharangao Taluka of Jhargram district. As we know, the bees play an important key role in sustaining our biodiversity and ecosystem. Also, they help in generating economic values. There are over 150,000 species of bees found across the world. Who visits the flowers, and bees have been dominant pollinators. Also, there are over 20,000 species of wild bees available worldwide. Of the 109 most important crop plants, no fewer than 87 species are entirely dependent on pollination by the animals. The main objective of this research is to identify the gut bacteria of Tetragonella independentis, since the microbes from the gut of animals can benefit their host by helping to digest food, improve malnutrition, and protect against the invasion of their pathogens, parasites, and diseases. For this, we collect the specimen, that is the stingless honeybee from the hives. Then, after that collection of these stingless honeybees, we dissect these bees to remove the gut in the control condition, that is the biosafety cabinet, and grow the culture into the agar and the broth. Later on, identify the various colonies over the agar plate by putting the control side by side so that the elimination is there. Later on, we collect the genomic DNA from this bacteria and put into the gel electrophoresis for the quality and quantity of the DNA, and then pass this to the sequence analysis and the results. So for the analysis of sequencing of this 16S RNA takes place, and for this sample is sent to the Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. Then the results are identified with the help of approximately 780 base pair fragments of the 16S rRNA gene was amplified from the isolates of the genomic DNA. The amplified fragments were compared to those sequences deposited in the gene bank database. The 16S rRNA gene sequence of the bacterial isolates displayed a similarity of 100% to the closest known species by using the blast at the NCBI. Based on the 16S rRNA gene, the isolate belonged to the phylum Fimbricatus, that is this bacillus species. On the basis of this, we classified and identified the 16S rRNA gene sequences collected from stingless bee worker. Operational taxonomic unit was assigned to the phylum level, and OUT, that is the operational taxonomic unit, was identified as Pristia genus. In the presentation, the sequence is shown and the blast result is also shown, so that the 100% similarity gives the evolutionary history was inferred using the neighbor joining method, and the isolates were aligned with closely related strains from gene bank, which is available on the NCBI site, and the accession numbers indicated in the tree also. And the basis of this, we conclude that the actually we isolate about 10 colonies. However, due to the time constraint and the economy, we evaluated only one colony for its 16S rRNA analysis. The common group of the bacteria found in Tetragonella independentis from gut is phylum Fimbricatus, that is the bacillus species, family Pristia genus, and the Arabactae is the species. Hence, we conclude that for the first time, we reported the Pristia arabatae bacterium from the gut of Tetragonella independentis from our region, that is the Dharangao Taluka of the Jhargram district. Thank you very much, madam.
So, what is the actual role of these bacteria in their gut?
The role of this bacteria is, madam, to digest the food material, actually, which helps to synthesize the honey in the gut.
Sir, actually, honey is not synthesized in the gut.
Yes, madam, it is synthesized in the gut and it is regurgitated by the honeybee into the pods.
I know they add certain enzymes, but
Yes.
if there are microbes there,
Mhm.
don't you think the honey will also have microbes?
Madam, not bacteria included into the honey, but during the digestion process, these bacteria secrete some enzymes, and because of this, the nectar is converted into the honey, and then this is expelled out from the body into the pods.
So, gut in the sense, which part are you telling? Intestine?
Yes, crop and the intestinal part, madam.
It was present in crop as well?
Yes.
Okay. So there was no difference between crop microbiota and intestinal microbiota?
Yes, yes, difference is there, madam.
Okay. Okay, sir.
Okay, nice presentation, Manoj Kumar. I would like to ask two questions. What is the purpose of isolating microflora from the gut of stingless honeybee? And one more question I would like to clarify is, have you utilized those microbial consortium for any other application?
Yes, madam. The purpose is that we are thinking that culturing of bacteria is quite easier than the other organisms. So, with the help of this bacterium, along with this, another some bacteria also, we want to synthesize the honey in vitro condition, that is in test tube or say in the flask, by putting nectar as a food to these microorganisms.
Okay,
This is
nice.
basic idea behind this.
Okay, nice.
Dr. Manoj Kumar sir,
Yes, madam.
the bacteria that you have just told about the gut microbes, so was this discovered before, or is it present in other animals also, or is it found only in this?
It is present other organisms also, madam.
Okay, huh.
That is why the data is present in gene bank in NCBI. So it is matched with this. That is why we say that it is a pistil arabectide.
Acha, that means it is found also in other animals,
Yeah, yeah.
other insects also.
Yes, madam.
Okay, but then this role is more important in the formation of the honey. Okay, yeah, thanks.
Thank you, madam.
Thank you, sir. We'll move on to the next presentation. The title of the presentation is Role of Microbes in the Bioremediation of Pesticides and Their Residues. The presenters are K.R. Padma, K. Adan, and Ramesh Arjun and Sankari. May I know who's coming? Who's presenting? Who's there from the group? I'll move on to the next presentation. AM mycorrhiza biofertilizer and mass manipulation for direct use. The presenters are Bumpy Ghosh ma'am, Devabritsa and Kamla Roy. Is it presented?
Madam, presented yesterday.
Okay, sir.
Physical mode.
Okay. So the next one, study of DOF protein sequence of different plants to understand the probable host plants of mycorrhizal association. Presenters are Kanwa, Samadatta Ghosh, Jangala, and Ayagari Ramlal, and Avika Samanta.
Yes, ma'am.
Yes, sir, you can start.
Myself, Kanwar Hasda.
Could you please turn on the camera if possible?
Yes, ma'am. Turn on.
No issues, you can proceed.
Myself, Kunwar Hasda, Assistant Professor, Department of Botany, Garbeta College, Midnapur, West Bengal. Before going to presentation,
Kanwar,
I will. Yes, sir.
বলছি
বলছি
ক্যামেরাটা
ক্যামেরাটা
খোলো
খোলো,
তোমার
তোমার
মানে
মানে
মোবাইলের যে
মোবাইলের যে
ক্যামেরাটা খোলো
ক্যামেরাটা
যাতে
খোলো,
তোমার
যাতে
ছবিটা
তোমার
দেখা
ছবিটা
যায়।
দেখা যায়।
এটা রেকর্ড
এটা
হচ্ছে
রেকর্ড
তো
হচ্ছে তো,
ফলে
ফলে
হ্যাঁ হ্যাঁ
তোমার
তোমার
মোবাইলের
মোবাইলের
ক্যামেরাটা
ক্যামেরাটা
অন
অন
করো,
করো,
অফ আছে,
অফ আছে, হ্যাঁ,
দিয়ে বলো।
দিয়ে বলো।
হ্যাঁ স্যার স্যার বলছি এক মিনিট
দিয়ে বলো।
দিয়ে বলো। পারছো না নাকি? ওইখানে দেখো স্ক্রিনে একটু টাচ করো, নিচের দিকে দেখবে একটা মাইকের মতো আছে, ওটা তো খোলা আছে। আর একটা দেখো ক্যামেরার মতো আছে, ওটাতে টাচ করো, খুলে যাবে তাহলে।
না
না পারলে ছেড়ে দাও, বুঝলে,
কন্টিনিউ
কন্টিনিউ করো যদি না পারো।
Yeah, he's
If not possible, continue. Huh?
you're muted also. You unmute, please.
Okay, okay. Before going to presentation, I welcome all dignitaries and participants. First of all, I would like to thank all the members of the organizing committee of this two-day national level conference, and thank you for giving me the opportunity to present my PowerPoint in this conference. My topic is study of dual protein of different plants to understand the probable host plants for mycorrhizal association. Mycorrhiza is a symbiotic association between higher plants, root and fungi. In this symbiotic association, plant easily absorb water and essential minerals from soil, and fungi receive sugars and nitrogen containing compounds from plant's root. The interaction of arbuscular mycorrhizal fungi and plant root in the rhizosphere is occurred through the stringolactones. Stringolactones play an important role in symbiosis. Stringolactones are signaling molecules that are
দেখো
produced by
হোস্ট
the root
প্ল্যান্ট ফর
of plants
মাইক্রোবায়োলজি
to form
অ্যাসোসিয়েশন, এটা
mycorrhizal
কোনটা
association.
মাইক্রোবায়োলজি করছি? কোন মাইক্রোবায়োলজি? অনেক তো মাইক্রোবায়োলজি
Stringolactone is
আছে।
the key component involved in the process of mycorrhizal fungal partner compatibility. A bioinformatics study has been done to find out a suitable host from a wild plant's mycorrhizal fungal partner. This process needs rigorous screening and time to study of the plants as well as fungal partner. The aim of the present study is that to find out the relatedness of different dwarf protein sequences of plant so that we can find the probable host plant of mycorrhiza collected from other species. Here, following objectives, some objectives are targeted from my research work. The common goal to develop mycorrhizal manure to collect mycorrhiza from wild plant, locally available, and find a suitable host plant root infect. Next, mycorrhiza fertilizer production technology involves finding a suitable host for the fungal species. This process needs a lot of time and skill. Third objective is stringolactone receptor, that is, dwarf 14 protein sequence are retrieved from the database and the homologs using BLAST in suitable host, that is local wheat, and track culture are done from rhizosphere, soil and root. Next target is pure culture of dominant arbuscular mycorrhizal fungal species from track culture. And fifth target objective is development of inocula from locally available plant, especially from weeds present in barren or contaminated land. And last objective is application of arbuscular mycorrhizal fungi inocula and treatment in barren land with suitable crop to find the growth and yield. So it can be concluded that dwarf sequence of the other plants that have the close similarity to the dwarf sequence may bind to the stingolactone and can be taken as a host plant for mycorrhizal association. This method can be a novel approach to find the host plants in a less time consuming and convenient manner. Thank you, ma'am. Thank you. Hello. Are you here? Hello, ma'am.
বাথরুমে গেছে তো Kunal, wait, huh? Wait. Name is coming.
Okay, sir.
Konkona Desh Mapalsa, Konkona De Mapalsaha, NG Muhajan, NG Chupada, NG Muhajan.
কি বললাম তো সে মাথায় ঢুকছে না ওখান থেকে বাই ফার্টিলাইজার চলে গেল পেঁয়াজ পিএইচডি গ্রাজুয়েট হবে এটাও গ্রাজুয়েট হবে কি ছিল তো বিনিতা কোথাও গেছে বিনিতা আসবে তুমি নেক্সট কেবল টিকে দেখে যাচ্ছে
Mirza ma'am, are you here? Mirza ma'am? Menuta ma'am?
Yes, sir. Yes, sir. So,
already completed.
Any questions? Any questions, or shall we move on to the next presentation?
ওরা বললাম বলুন তো ওরা বলুন
The topic of next presentation is microbial and physicochemical analysis of Fakir Kuwa water at Medinapur town, Paschim Medinapur, and its current potability status. The presenters, Shamaita Aicha, and please pardon me if I'm pronouncing it wrongly. It's Dulal Kumade and Bratati Banjara.
Yes, ma'am. Please.
I'm sorry if I pronounced your name wrong.
Bratoti Bhanja.
Okay. Please, Bratati, you can start. You can start.
Samaita. Okay.
Yes, ma'am. Yes, sir. So I have done this work as my MSc dissertation in Midnapur College Botany Department. So Fakirkua is a very famous well of Midnapur town, and local people believe that the water has some magical healing properties, and they consider this water as a holy water. So we decided to scientifically test the water quality because local people drink the water directly from the well without any basic purification treatments. We have performed all the experiments in 3 different seasons of this year. We have done MPN testing using macroamphibian broth. It is a selective and differential media for coliform. The reason we are choosing coliform bacteria is that they are largely present in human intestine, and they also naturally present in human or other warm-blooded animals' fecus. So the test result was positive, and it means that the fecal contamination has been occurred in the water, and we compared the result with the standard value given by APA, American Public Health Association. According to the index, the water has medium risk for drinking. That means we should not directly consume the water from the well. We also have done selective media plating. EMB, eosinophilic blue media is selective media for E. coli, and E. coli colonies were grew in the 3 different months of this year, and Staphylococcus and Pseudomonas colonies also grew on Staphylococcus and Pseudomonas selective media. Both of them are highly pathogenic bacteria. We have also done chemical testing for arsenic, iron, copper, sulfate. All the parameters were normal in range. So the cause of contamination maybe is from the nearby septic tanks leakage or defaulty sewage system present in the locality. So we should aware people about the condition of the well and try to prevent them from further consuming the water. And further study is also required to needed more details about the water. So people of Midnapur town has many religious sentiments and beliefs. but we should try to scientifically aware them about the current situation of the rainwater.
Any
And
questions?
complete.
Yes, yes. Questions? Thank you, Samaita. I'll move on to the last presentation. Survey of disease affecting banana and paddy crops growing in Pingla and Sabang blocks of Paschim Medinipur. And the presenter is Koninika Sen. Are you there, Koninika? Koninika?
Madam
Madam
Konika may be
Konika may be
absent.
absent.
May be absent. So that's the end of today's presentation, sir. So I think we have come to the end of this today's day 2 presentation. As we conclude, on behalf of the organizing committee, I would like to extend a heartfelt thanks to all the panel members for such an engaging discussion and valuable insights. And thanks to all the presenters for presenting your research finding. And special thanks to Devabrat sir for organizing such a meaningful conference and also providing such forum to all the researchers to share their findings and also for creating such inspiring scientific spirit in them. Thank you very much. It's almost 10:30. It shows that the spirit is on. That's why we all are here till 10:30. Thank you, sir, for providing this great opportunity to all the presenters and as well as bringing us to witness all this.
Thank you,
Thank you,
madam,
madam,
for your
active
active
participation.
One
One
announcement
announcement
to
to the
the presenters,
presenters,
send your
send your
PPT or video
video
PPT
PPT
or
or
both
both
to
to
our mail.
our mail.
We'll provide
We'll provide
the
the
drive link.
drive link,
And
and
shortly we'll
shortly
prepare
we'll
the
prepare
list
the
those who
list
have
those who
already
have
sent
already
us,
sent us,
and
and
those
those
who need
who
to
need to
modify it
modify
again
it
send
again
it.
send it.
And
And
second, we'll
second, we'll
provide
provide
link for feedback
link for
through
feedback
email. So
through
don't
email.
worry
So
regarding
don't
this.
worry regarding this.
Everybody will
Everybody
receive
will
the mail,
receive
and
the
through
mail,
mail
and
we'll
through mail
provide
we'll
the
provide the
Google
Google
form
form
for
for
feedback.
feedback.
And prepare
And
your
prepare your
full
full
papers
papers for
proceedings
proceedings
for
for
publication. Okay,
publication. Okay,
but it will
but
take
it will take
2 months or
2 months
more.
or more.
Already yesterday
Already
I
yesterday
discussed
I
with
discussed with
publisher,
publisher,
and they
and
told me,
they
sir, you
told me
prepare
prepare
everything
everything
in a
in a
nice
nice
manner so that
manner
I
so
can
that I can
publish it
publish
within
it
in 2
within
days.
2 days.
So we have to
So we have
prepare
to
it
prepare it
through
through
rigorous
rigorous
review
review
and
and
rigorous
rigorous
editing works. And
editing works,
during that time,
and
your active participation
during
is
that
required
time
because
your active participation is required
many
because
more
many
willing to publish
more
their
willing to publish
papers. It may be research or
that
review work for
paper,
short
maybe
notes.
research or review work or short
During
note.
your
During your
presentation, that means
presentation,
during your sending
that means during
article,
your
you
sending
send one
article,
good
you send
photograph
one good
zone so that
photograph
you can
zone so that you can
amalgam all the
amalgam all the
photographs
photographs
in two
in
plates.
2
Okay.
plates. Okay.
And
And
third is
third is
e-magazine.
e-magazine.
We'll
We
going
will
to
going to
publish on
publish on
e-magazine,
e-magazine,
which will be
which will be
published very
published very
soon.
soon.
So
So
convey the
convey the
message to
message
your
to your
students, to
students,
your
to your
friends, to
friends,
your
to your
colleagues, so
colleagues,
that they
so
can
that they
send
can
the
send the
write up. Okay,
write
so
up.
that
Okay, so that
we can
we can
publish it in an
publish it in an
e-magazine format.
e-magazine
But
format. But
we'll provide
we
time
provide
for
time
that 2
for
months.
the
In the
2
month
months.
of
In the
March,
month of
we'll
March, we will
release the
release the
e-magazine.
e-magazine.
In the
In the
e-magazine, we'll
e-magazine, we will
take the
take the
article from
article
different
from different
teachers,
teachers,
students,
students,
researchers, scholars,
researchers, scholars,
etc. on the
etc.
basis
On
of
the basis of
interdisciplinary approach
interdisciplinary approach
on plant
on
animal
plant animal
and
and
microscience. Okay.
microscience. Okay.
So
So this is my
this is my
message
message to
to
all
all
I also convey my thanks to all resource persons, professors, teachers, scholars, students who gave their best to make it complete. Thank you again. Good night.
Good night.
Giving us this great opportunity.
Thank you, sir.
Thank you, sir. Good night.
Thank you, sir.
Thank you. Thank you, sir. Good night.
Thank you, sir. Thank you, Ananda. Thank you.
Thank you, sir.
Thank you,
Thank you,
madam.
madam.
Good night.
Good night.
এটা একটু সেভ করতে হবে কোন ঘন্টা করবো
Thank you, sir.
থ্যাংক ইউ ম্যাডাম
Thank you, madam. Thank you.
কোনটা এইখানটা ক্লিক করবো স্টপ